this one's okay.
1, 2. 1, 2. Testing 1, 2. 1, 2.
Okay, I think we are ready for this morning. Hope everyone slept well. I'm Tom Walker from the University of Warwick, and I will be chairing the first few talks this morning on microsporidia. So first up, we have Jeremy Heron from ASIPE here in Kenya, and he's changed his title already, but his title is now symbiont-based transmission blocking for malaria. Jeremy.
Okay, is that working? Yeah. Okay, great. Okay, great. Thank you, Tom. It's a real pleasure to be here, to see everyone again, and to give you a bit of an update on our work on microsporidia. So just by way of a very broad introduction, we're interested in symbionts. As many of you will know, microbial infection of a host doesn't always result in disease. We're often more familiar with pathogenic microbes, or most of the world is. But actually, there's a lot of really interesting microbes on the other side of that sort of spectrum. Things that are mutualist in sort of the yellow area, and true sort of symbionts in the green. looking at these infections in mosquitoes, there's sort of some trends that we see with those microbes that tend to be more symbiotic in terms of where they are in the mosquito. On the pathogenic end, you get these kind of disseminated infections that kill your host relatively quickly. A lot of mutualists, I think, are kind of in the midgut and on the cuticle, which have maybe more mild effects on their host. And then on this sort of more symbiotic end, you get these associations which are more long-term. In a lot of cases, the symbiont is actually inside the cells of the host, and I put some eggs that are infected there to show that vertical transmission is something often associated with symbionts. Another difference is how these behave at sort of a population level. Things that are pathogenic, you tend to get this sort of predator-prey type cycling. The pathogens are sort of a victim of their own success, killing a lot of hosts, and often are found at quite a low level in the population. On the other end, symbionts, because they don't actually kill their host, can spread more effectively and actually reach much higher prevalences in the population. So we're very interested in things more towards that end, and specifically in the context of their role for transmission blocking. This is where we do a lot of our work in Western Kenya, semi-field insectaries with labs nearby, so we can bring mosquitoes in from the field, and pretty much everything I'll discuss today is done on F1 mosquitoes. This was originally done because we needed to maintain the microbiome and the symbionts from the field. A lot of times, if you keep mosquitoes for many generations in the lab, they lose a lot of their interesting microbes. So in that sort of setup, we found this interesting new Microsporidia found in Anopheles mosquitoes, which is Microsporidia MB. Microsporidia, just in general here, are spore-forming obligate intracellular eukaryotes, distant relatives of fungi, a very ancient branch. They don't really have many of the features we associate with fungi. They do have a very complex life cycle, a lot of different transmission patterns, many different hosts, different phenotypic effects. I don't have time to go into all of that. But specifically now, Microsporidia MB. So far, we've only found this in Anopheles mosquitoes. We found that it has very high infection intensity. The picture on the right there in red is actually Microsporidia inside a developing Anopheles egg. So loads and loads of Microsporidia cells inside mosquito host cell, but a very minimal effect on the host fitness. It's intracellular and is found in specific tissues and organs in the mosquito. It's vertically transmitted from mother to offspring, and recently we've also shown that it's sexually transmitted. So those are a suite of characteristics which we think are quite interesting, and that's why we've really taken this symbiont forward and done more study on it. So I'm going to talk a bit about transmission blocking of Plasmodium, which is a key characteristic of interest in our work. Basically, I'm sure everyone's very familiar with the Anopheles mosquito life cycle, going from an adult mosquito that's blood-fed, laying eggs, going into larvae, then you have mating between males and females, and the cycle completes itself for Anopheles gambiae and arabiensis. That's usually around 14 days. But importantly, it's about 3 to 4 days from the blood meal to laying the eggs. So that then contrasts a bit with the Plasmodium life cycle, which actually is quite long relative to the mosquito life cycle. Mosquito takes an infected blood meal, then the Plasmodium parasite develops, goes through a number of stages in the midgut of the mosquito, eventually entering into the enterocytes, forming oocyst and then sporozoites. So when we're looking at transmission blocking, we can investigate various stages to sort of see where transmission blocking is occurring, and that will be relevant for some of the things that I'll show you next. So the original experiment we did with Microsporidia MB was to take mosquitoes that were infected and uninfected and feed them on blood from donors that had gametocytes. In this case, we looked for sporozoites, so the very last infectious stage of Plasmodium in the mosquito, and we found that there were no sporozoites in any of the mosquitoes that had Microsporidia MB. So a strong blocking effect, but that didn't really give us that much information as to where the blocking was occurring. In these same mosquitoes, we also looked for oocysts, and we also didn't see any oocysts, so suggesting that the blocking effect was happening actually prior to the formation of oocysts. So that's where we were a few years ago. We've now been building on this work, and I'll show you some new data from the field. Because we work on F1 mosquitoes, we go to the field a lot. Actually, we've been going to our main site 3 days a week for the last 2 years. And we collect a lot of blood-fed mosquitoes, bring them back to the lab, incubate them for about 4 days, let them lay eggs, and we use those eggs for a lot of our experiments. And so we had actually over 20,000 mosquitoes, but not all of those were Microsporidia infected. We had about 3,500 that had Microsporidia. We then matched those to uninfected control mosquitoes that were collected on the same days, and we screened both of them for Microsporidia and for Plasmodium. So this is looking sort of somewhere in the middle of that incubation period of Plasmodium. And what we found was quite a strong effect. So the proportion of these mosquitoes that had Plasmodium was higher in almost every month in those mosquitoes that didn't have Microsporidia. And a number of mosquitoes that had Microsporidia also had Plasmodium. We think that this is sort of showing the transmission blocking in action. Looking at the densities of Plasmodium in those with Microsporidia and without, we see that the densities were much lower in those that had Microsporidia. So it looks like this blocking effect is happening in the field, which is really interesting and important because previously we only showed it in the lab, although with patient blood. And it also looks like it's happening quite early on in the process. So that's all great. You see that there's a strong blocking effect. We know that Microsporidia is a naturally occurring microbe. So why do we still have lots of malaria and Plasmodium in the field? Well, the answer to that is that there isn't really enough Microsporidia to be having a major impact. So that's something we wanted to get a better handle on, how much Microsporidia is there naturally in various sites across Kenya where we do a lot of our work. Postdoc in the lab, Oscar, who has a poster on this, so I won't touch on it too much, did a transect across Kenya looking at the levels of Microsporidia and saw that there was quite an interesting range, but generally the highest sites that he found were about 10 to 12%. Interestingly, Oscar also screened males. This is data for females and larvae, and found that the amount didn't always line up. So it's clearly quite a complex system. We also have this one site, sort of our backyard site, where we go weekly. And so we've got really good data from the last 2 years looking at the levels of Microsporidia there, which seem to fluctuate around 10%. So overall, generally around 10%, but we've seen levels as high as 60, and in some cases we don't see Microsporidia. So how would this all be useful? What are we kind of thinking? The idea is sort of to strategically disseminate Microsporidia, increase the levels from a basal level somewhere around 10% to a level that hypothetically would protect that population against Plasmodium infections. So we would be releasing in areas where Microsporidia would do well, and maybe at times when there are not many mosquitoes, that would increase the levels and decrease the levels of Plasmodium. Theoretically, it would work. I think my colleague Prashant is going to share some modeling data with us in his talk coming up. but there's a lot of information that's missing to see, how effective this would be. And really, what's important is understanding how microsporidia could spread. And so, a student, Naomi, who's also got a poster today, is trying to understand, to model the potential spread of microsporidia. And this has been a really useful exercise for the whole lab, working with modelers, because it kind of gives us an idea of what are really the key parameters that we need to study in the lab and understand better. So things like transmission rates, survival of mosquitoes infected and uninfected, mating success, development time. So this really gave us, a key sort of roadmap for what we needed to study. And that's kind of an overview of all the work that's going on. And a lot of these are represented by students that are presenting either after me or have posters. So basically, we're really interested in studying microsporidia transmission. That seems really key. Maternal transmission, but also horizontal sexual transmission. We've investigated paternal transmission, which that's transmission directly to the offspring from a male, and also environmental transmission. Cynthia has a poster on that. So far, we don't really see any environmental transmission, which is interesting. We see all these fluctuations in the field. We're really trying to understand, to get a better grasp of what's driving those fluctuations. So looking at the environmental conditions, how those affect microsporidia. I'll talk about a few of those in the talk, and others will be covered by colleagues in the lab. And then looking a bit, in detail on the host-symbiont interactions, the fitness effects, which are obviously very important. And then Jackie had a poster looking at effects of microsporidia on host gene expression. So a little bit on transmission. A number of different possible transmission routes, which are shown in that diagram. So far, we've not found that all of those do take place. I'll go through a few of them. So the key, or the sort of primary route of transmission, I say primary just because it's the first one we found. We don't know if it's the most important, but it's maternal transmission. Microsporidia has pretty high maternal transmission rate of, on average, about 78%. It's a bit low relative to something like Wolbachia, which is 100%, but it's still appreciably quite high. We've got some really interesting results about how that transmission is happening in the ovaries, and Thomas, postdoc in the lab, will discuss that in the talk coming up. Paternal or sexual transmission, actually this is sexual transmission, is also seems to be an important route of transmission. There's more effective transmission from males to females, but there is also transmission from females back to males. We're also really interested in how this happens, and got some nice microscopy that you'll get a bit more detail on, but you can see in this picture that microsporidia is actually in the testes of these Anopheles mosquitoes, and then gets sent out with the sperm cells. And you can see these microsporidia full cyst-like cells that are all packaged in with the sperm. Okay. Environmental transmission. We looked at where microsporidia is in a number of tissues, and those are the graphs. Just sort of to summarize that, it seems to be really primarily in the gonads, but we did see appreciable levels of microsporidia in the midgut, which led us to sort of explore the possibility of a kind of fecal-oral transmission route, but that really doesn't seem to be happening. So we've so far not observed any environmental transmission. Moving on to environmental factors. So here we can study these in two ways. I don't know if that's what we can do about that. Okay, kind of looks like it's a bit stable now. Okay, so looking at environmental factors affecting Microsporidia, we've done some experiments in the lab that I'll talk about, and then there's also some field data which Joseph, who's talking after me, will talk about. In the lab, we've looked at temperature and nutrient availability. In terms of temperature, Microsporidia prevalence is actually goes up at higher temperatures. So if we maintain lines at temperatures of up to 37, the furthest on the right there, we have the sort of complete infection of all mosquitoes with Microsporidia, whereas at lower temperatures, we get sort of a higher level of uninfected mosquitoes in lines. And these are lines which have been isofemale lines, but then split amongst all these different temperatures, so all from the same female. In terms of the density, we also see a higher density of Microsporidia at the higher temperatures. But an interesting finding here was that at the highest temperature of 37, we saw a drop in the survival of mosquitoes with Microsporidia relative to those that didn't have Microsporidia. So a potential fitness cost that is coming out when you have a really high intensity infection at a high temperature. Nutrition, we raised larvae on a really low diet, sort of a starvation type diet, and a normal diet. The prevalence of Microsporidia was a little bit lower in the low diet, but not a huge difference. And we see that the density of the infection was actually also lower on the low nutrient diet. So when you starve mosquitoes, they have slightly lower levels of Microsporidia. And so now moving on to other fitness effects. So again, just we're going to look at different parts of the life cycle of mosquitoes. So that's why I put that there. In terms of the number of eggs laid, we've seen no difference between Microsporidia infected and uninfected. In terms of the hatch rate, there's also no difference. In terms of the survival of larvae, we've also seen no difference. And even if we maintain mosquitoes on a really low diet, where only 20% of the larvae actually make it to pupae, we still see actually no difference between those that have Microsporidia and those that don't. This has been really the strongest fitness-related phenotype. Microsporidia infected mosquitoes develop faster by about a day and a half, which is seen across the board, really. We've even seen this in Anopheles gambiae as well as arabiensis. Most of the experiments we've done are in Anopheles arabiensis. This effect actually goes away on a low nutrient diet. So the high nutrient diet histograms are on the left, and on the right, the low nutrient diet, it's no longer significantly different. So a lot of the questions are, if they develop faster, is it just that they are smaller? That's sort of known to happen in a lot of cases when something, an infection goes through larvae. That's really not the case. So here we looked at the size of males and females. MB positive females are the same size as MB negative. However, the males are actually significantly bigger in the MB positive. Again, that effect goes away on a low nutrient diet. Effects on adult survival. Microsporidia infected mosquitoes survive significantly longer if they're maintained just on sucrose solution. And if we starve adult mosquitoes, just keeping them on water, Microsporidia infected also survive longer. In terms of mating success, so this is looking at males infected with Microsporidia. Here we're looking at stamina. In this experiment, we put one male with 3 females, allowed them to mate for 3 days. If they were still alive, they got transferred to a new cup with 3 fresh females. What we see is that the Microsporidia infected males kept going much longer, so 60% of them made it to the second batch of females, whereas only 14 of the MB negative. And overall in this experiment, the number of females mated was significantly higher in the males, 1.45 versus 1.29 for MB negative. Another experiment to look at head-to-head competition, where we put 10 males infected and 10 males uninfected together with 20 females. We used a dye to mark those that are infected. in 3 replicates of this experiment, and in the other 3 replicates, we marked the uninfected males to rule out any effect of the dye. And in this case, we had 70% of the females were mating with Microsporidia infected males overall. So a really strong effect in both of those lab-based experiments on mating. So sort of just to conclude, I've given you kind of a big picture. We're trying to really understand all these different aspects of this Microsporidia system, and what factors are contributing to Microsporidia spread and what factors are decreasing levels of Microsporidia. So I've shown you that vertical transmission is good, but it's not perfect. We're losing quite a few mosquitoes every generation. We make up for that by horizontal transmission, and also to some extent paternal transmission, where the male infects the offspring directly. Temperature has a little bit of a complex effect on Microsporidia. Nutrient availability seems to decrease the levels of Microsporidia, and other environmental conditions are kind of positive and negative, and Joseph will talk a bit about that. All the fitness effects we've observed in the lab seem to suggest that Microsporidia infected mosquitoes have higher fitness, but we're continuing to investigate others. So I think that, that's all. Just to acknowledge everyone that's done all this work. We've got a really excellent team of staff and students that have done this all, and to acknowledge the funders, and I'm happy to take any questions. Thank you.
Thank you, Jeremy. Questions for Jeremy?
How would one go about How would one go about releasing Microsporidia? Is it by releasing males that are infected with it?
Yeah, I mean, that's something we are considering. At this point, yes, because it's transmitted sexually and males obviously are less dangerous, the idea would be to release males.
Tony.
Thanks, Jeremy. I'm curious about the vertical transmission. So when you did the temperature experiment, you mentioned that those are offspring coming from an isofemale line. Is that right?
Mm-hmm.
And so at the lower, was it the lower temperature, they did better, or vice versa? Which way around was it?
They did better at a higher temperature.
Better at higher right. But at the lower temperature, in the lower temperature, there were very few that actually established the Microsporidia.
So in that experiment, basically, you take a female that's infected, you split her offspring into 4 different temperatures, and you do that for many different females.
Okay, so what I'm getting at is that presumably they all got a dose of Microsporidia, and then they lost it transiently.
Yeah. So when we talk about vertical transmission, we're talking about transmission from one adult mosquito to the next.
Yeah.
What's happening between that, we don't fully understand. But it seemed that possibly all your eggs get it, but some of them lose it over the course of larval development. That's what that
must be what
data
happened,
would
right?
suggest.
Because they all got it.
They
all could get it, and in some cases all got it. So they must have all had a dose at the start.
Yeah.
And at the lower temperature, they must have had a dose.
Yeah, that's what that suggests.
Any other questions? Okay, I've got a bit there.
Hi, really interesting work. So when looking at the infections from the field mosquitoes, are you only looking at presence or absence, or are you also looking at density? And if you're looking at density, does that also have an effect on Plasmodium infection? Like, is there a minimal threshold?
Yeah, I think that's something we need to investigate further. We don't know if there's a minimum threshold for that, but the ranges of densities we see in the field are relatively wide, and we do get some mosquitoes that have a very low density infection. It could be that those are the ones that have maybe acquired it through mating, and that if you acquire it through maternal transmission, you have a higher density. So yeah, we do see some with a low level that maybe aren't protective, but we don't really have enough data to show that.
Thank you.
Thank you. Any more questions for Jeremy?
Thanks. Could you comment on the prevalence differences between different larval sites, sort of more permanent versus more temporary? What's the latest on that?
Yeah. Oscar's got a poster on that where he's looked at irrigated and non-irrigated sites. And I think we tend to see higher levels in irrigated sites. It might be that where the density of mosquitoes is higher, we tend to get higher levels of microsporidia. But I would say we don't really have enough data to say that conclusively, but there is a trend towards that.
Okay, I think we're running a little bit late, but we're switching over the projector. Is that correct? Before the next talk. So I might as well ask one, Jeremy. Sorry. Given that it's environmentally acquired, can you see microsporidia in other mosquito species outside Anopheles genera in the same breeding sites? Do you see it in Culex or Aedes?
Well, we don't see any environmental acquisition, actually. Okay. So the only routes that we've found, I mean, we can't rule them out, but so far we've only found sexual transmission and maternal and paternal transmission. So what we've tried is, all sorts of feeding experiments, grinding up mosquitoes, even microinjection, like thoracic microinjection,
And you can't
doesn't transfer it. So it seems to have a very specific route of infection. Yeah. And going on from that, which is not too surprising, we don't see it in any other organisms from the larval sites or any other mosquitoes. We've only seen it in Anopheles so far. Yeah.
Fantastic, thank you.
Yes.
Might as well ask him lots of questions.
What about?
Yes.
Thanks for the talk. Super interesting. What about spores? Have you been able to isolate spores from Microsporidia MB?
No, we've not seen a Microsporidia MB spore, but if it didn't make spores, it would be the first Microsporidia to not make spores. So I presume that they exist.
That's super interesting.
The only place we think would be the best place we think to look for them would be in the male reproductive tract, because that's probably where they have to exist outside of a cell. But even what we see now, because we're staining with fish, which doesn't stain the spore stages, and we see it lighting up. So even in the testes and the vas deferens of the male, they seem to be still inside a cell. But how they get from that into the egg, whether their spores are formed at that point, we don't know.
That's so strange. Thanks.
Yeah. That was the testes. But Thomas is giving a talk, and he'll show a bit of it going actually down into sort of the ejaculatory duct and all that. And it's still at that point isn't in a spore form, but it may be that it goes at some point between there and getting to the egg, it may convert into a spore for a very short period of time. But I think we looked at the genome. Lillian's talking about that. They do seem to have spore proteins. We've detected them in the spermatheca by PCR, not seen them in the spermatheca. Yeah.
Right, I think we're going to have to move on. We're sticking with the projector, apparently, so well done, Jeremy, for filling all that time with great answers to the questions. So we have now Thomas Achuru from Jeremy's group in Esipe, and he's talking on vertical transmission of microsporidia in Anopheles gambiae, and it occurs through the germline stem cell infections. I'm not going to tell any jokes, don't worry. All my jokes are not appropriate for this kind of scenario.
good morning, everyone. That was a short break. I hope you had time to have a seat on your desk. So yeah, I'm going to zoom into one aspect of Jeremy's talk, and that is vertical and horizontal transmission mechanisms of Microsporidium B in Anopheles arabiensis mosquitoes. My name is Thomas Onchuru, a postdoc with Jeremy. So as Jeremy mentioned earlier, we discovered Microsporidium B in Anopheles arabiensis as a native symbiont. Jeremy has shown that Microsporidium B infected mosquitoes cannot transmit Plasmodium parasites because they don't develop. And we know that there is vertical transmission from mother to offspring of Microsporidium B, and also horizontal transmission from males to females and females to males. Another aspect is that Microsporidium B acquired from males during mating can be transferred to the offspring, and that is paternal transmission. Now, we know that these transmission mechanisms are happening, but we don't know the mechanisms behind this vertical, horizontal, and paternal transmission. And that's what my talk is about, to try and understand how these transmission routes are taking place. So why is it important for us to study mechanisms of transmission? As Jeremy mentioned, the end goal of this project is to try to introduce Microsporidia infected mosquitoes in the field and expect that they will mix with wild populations and spread and sustainably maintain Microsporidium B in these populations. Now, transmission will play a key role in the spread and maintenance of Microsporidia, and therefore it's very important for us to understand how this is happening. So basically in my talk, I'll focus on transmission mechanisms from the female to the offspring, but also I'll touch on male to female transmission. So to study vertical transmission, that is from mother to offspring, what we do is we take an infected mother, we dissect out the reproductive organs and mainly the ovary and the spermatheca, and then we subject this to microscopic examination to know exactly where Microsporidia is located. And we use a fish technology to locate where Microsporidia is located. And throughout my talk, you will see a very strong green signal that is just a general DNA stain that shows the cell structure of the host, but also for Microsporidia, but also a very specific probe that only attaches to Microsporidia. So whenever you see red, that is Microsporidia, green, that is general DNA. And as expected, when you stain an ovary that is not infected with Microsporidia MB, you will only see the general DNA staining. But when you stain with Microsporidia, you stain a Microsporidia infected ovary, you will see this. Sorry, this is quite fancy, but you will see a strong red signal in some places within the ovary. And this was first evidence that vertical transmission is happening when Microsporidium B actually infects the developing ovaries. And interestingly, if you zoom into some of the regions, you will see that this is kind of a developing egg that is full of Microsporidium B, and this shows that Microsporidium B actually gets into the developing eggs for it to be transmitted to the next generation. Now, the ovary is a large organ. It's made up of small subunits called ovarioles. Thank you. And those ovarioles have three regions. That's the primary follicle, which is going to be the first egg. We also have the secondary follicle, that will be the secondary egg. And then we have the germline stem cells, where these eggs originate from. And therefore, we also wanted to see exactly in this basic unit of the ovary what is happening. And when we stained with Microsporidia specific probes, we see that Microsporidia is present in the germline stem cells, in the secondary egg, and in the primary egg. And that pointed to the fact that it gets into the basic unit of the ovary. And actually what we see is that some of the first eggs, so if you look at these two, one has Microsporidium B, the other one does not have Microsporidium B, which points to the fact that probably the Microsporidia is originating from the stem cells. Once those cells divide out of the stem cell region, they get their share of Microsporidium B, but some do not get Microsporidium B. And actually that kind of gives us some information why we never see 100% perfect vertical transmission, because some of the cells are missing their share of Microsporidium B from the germline stem cells. Now, the ovariole goes through a series of developmental stages from stage 1 up to stage 6. And these are characterized by the change of size of the first egg, but also the proportion of the oocyte within these eggs. And now we wanted to know what is happening exactly in every stage of this development. And what we see is that in each and every stage, Microsporidia is present in the main egg, the secondary egg, and the germline stem cells. And actually what we noted is there's kind of a trend, there's an increase in the amount of Microsporidia MB. We actually wanted to find out how much Microsporidia is in these different sections of the ovariole across the different stages. And what we see is that for the primary egg, the germline stem cells and the secondary egg, Microsporidia begins at relatively high amounts. It increases in the second stage and kind of plateaus. Interesting for us, we found that in the first egg that is just about to be laid, Microsporidia MB increases gradually as the different stages increase. Now, more interesting, this coincides with blood feeding. We know that blood feeding is very important for egg development. When mosquitoes emerge, egg development is arrested until those mosquitoes have a blood meal. Now, after a blood meal, then egg development resumes. And in this case, we see after blood meal, Microsporidia also increases. Now, that shows that Microsporidia development are synced with the host developmental cycle, such that it stops growing when there is no blood meal, and then after blood meal, it gradually increases. That is very interesting for a symbiont to sync its development with the host development. And this work agrees with Edward, a PhD student who's presenting next, who sees that after a blood meal, the high density infections increase within the host mosquito. Now, we also have more evidence that there is active cell division of Microsporidia within the developing ovaries, as you can see here. Yes, here. Here, yes. There are some cells trying to detach from each other, showing that there is kind of a separation from one another. Now, that is happening within the host cell. And we also have evidence that within the ovary, it supports the different developmental stages of Microsporidia, because we can see different sizes of Microsporidia within the same ovary, pointing to the fact that this region supports the different developmental stages. Now, with so much Microsporidia in the host cell, you could expect that there is an effect on the host. If you talked to Jacqueline yesterday in our poster, she must have informed you that within this cell, the ovaries, we don't see a differential gene expression when Microsporidium B is present. And this kind of points us that this symbiosis is very well established, that it doesn't cause any consequences to the host. We've done similar work in males, whereby we've also isolated or dissected out female reproductive organs, and we've stained them for Microsporidium B. What we see is that Microsporidia is mostly located within the testis and is largely present from other reproductive organs such as the accessory glands. And more importantly, within the testis, we see that microsporidia forms kind of clusters. Now, at this stage, this is towards the tip of the testis. This is where you see spermatocytes before they differentiate to form sperms. And it seems that microsporidia earlier on infect the spermatocytes, replicates within those spermatocytes, and this is what is transferred along the tube that connects the testis to the ejaculatory duct. This is the first vas deferens that connects the testis to the ejaculatory duct. And microsporidia, we see most of these clusters swimming together. We never see them swim, but they are located together with the sperms in these tubes ending in the ejaculatory duct. That means that microsporidia just follows the sperms and waits when that male is going to mate with the female, and then it is transferred to the females. So in conclusion, we have seen that MB infects and replicates within the ovarioles, and more importantly, we see it in the germline stem cells. Now, that is a region that is very well controlled. Nothing gets in there by chance. And it seems that there's kind of an intimate association between the host and the microbe that allows it to be vertically transmitted this way. We have also seen that across the different ovarial stages, microsporidia is present, and that is a stable infection. But more importantly, the presence of microsporidia in males points to the fact that we can use that as an effective and acceptable dissemination strategy, because males don't bite and therefore they can be easily accepted by the communities where they are released. And lastly, I'd like to thank the entire team, the SymbioVector team that supports this project, the funding agencies of the project, but also the ICIPE co-funders who indirectly support this project. And also thank you for your attention and patience with the projector. Thank you.
Thank you, Thomas. Great name. I think in the interest of time, we should just move on. And if you don't mind taking questions during the coffee break, is that okay? We're sort of really behind time now. So we've got the next speaker is Edward. Also for Jeremy's Edmund Makula. You okay? Okay. We're doing a change of projector now. Yeah, would you like to take any questions, Thomas? Because you've got time now. We're going to be here till about 10 p.m., aren't we? Do you want to go up to the stage and we'll give you, if you get your microphone. Any questions then for Thomas? Yes, Penny.
Hi, yeah, I was really interested in the, the effect of the blood meal on the microsporidian growth, and I was wondering then if you see any relationship with the age of the mosquito and the density of the microsporidian infection.
I will defer that question until he gives his talk. He's addressing that. Okay. Yes.
Any more questions? Tony.
Yeah, because we have some time, I mean, I asked the same question that I asked to Jeremy.
Yes.
So, apparently they all get a dose of microsporidia. If it's in permissive temperature, all of the offspring are getting microsporidia. So, do you agree? Is that reproducible?
I mean, no. So the simple answer is no. The first eggs, the batch that is laid, based on microscopy, not all will have microsporidia. Some will have microsporidia, some will not have microsporidia. So it seems that not all eggs receive microsporidia. But as Jeremy said, we don't know actually what happens between the egg is laid, the hatching, and larval development. Probably some are lost at that point, but we don't know at this point.
Some are lost, but also some might acquire it then, because if some of the eggs are laid without microsporidia, yeah, at a permissive temperature, all of them can get it, then are they acquiring it from their siblings? Or
So far, what we've done with environmental transmission, we have not seen any. I mean, so Cynthia has seen that some can be acquired by larvae, but it's very low amounts. So it's possible that they acquire it from the environment, but what we've seen, it's under low amounts. Actually, yes. Yeah, so they are not sustained within the mosquito for a long time.
Okay. Are we getting closer to going? I think so. I think you can sit down, Thomas. We'll give you a break. Okay, we have Edward Makulu, who's going to talk about microsporidia, abundance and intensity in germline and somatic tissues.
Okay. Thank you for the introduction and good morning, everyone. Welcome to my presentation. The title has already been highlighted, so I think I'll just go straight to the main issue. So, okay. So since I can call it the discovery of microsporidia, various important information has been highlighted or obtained from the discovery, and one of the information is that it impairs plasmodium development in the mosquito vector. It is vertically and horizontally transmitted. It is in major malaria vectors, and it's a naturally occurring endosymbiont that is showing capabilities of true endosymbiont characteristics. But what does this mean? This means that microsporidia sits at a very good position to be used as a strategy for control and management of plasmodium transmission. However, for an effective microsporidia MB-based transmission, effective microsporidia MB-based malaria control strategy, two important things must be highlighted. Those things Thomas has already highlighted, which is the efficiency of the efficiency of transmission, and also the enhancement of host fitness. And together, when you combine the two factors, is that you get an improved spread and maintenance of microsporidia in a population, something that is important for it to be used as a control strategy. So for us to achieve these two, it means that it is important to understand where the symbiont is located and what is the tissue levels of the symbiont in these tissues, and this is what we set on the journey to investigate during my PhD. So before we can move on, is that it's also worth to note that in the subsequent presentation, I'm going to talk about quantification, and generally we did two types of quantification, which was relative and absolute quantification. This was generally because the host has always a change in cells, which may be important to highlight what type of quantification we did. But in the presentation, I'll be measuring only on the absolute quantification. So most of the figures that I'm going to present is be on the absolute quantification. So for starters, is that microsporidia, we could localize it in the somatic and the germline cells, as you can see from the results. And it was also worth noting that the tissues that showed high levels of microsporidia were the germline cells. Were the germline cells, which is the ovaries and the testes. The ovaries and testes were the highly tissues infected with microsporidia. But it's also worth noting that we could see that there was low levels of microsporidia in the fat bodies. Other species of microsporidias that are pathogenic are usually having high levels in the fat bodies, because these are the tissues of main infections. So the fact that microsporidia is maintaining those levels, it means that it's like regulating itself so that it cannot have a high effect or of host fitness on those on the host. Also, we localized microsporidia, something that we did with Thomas, the postdoc, and I'll not really go much in that work because he has highlighted most of the work. We could localize it in the developing oocyte, something that he has highlighted, and also we could develop it in the maturation and the transformation stages of sperms in the testes, something he has also highlighted. But one thing that he has not really expounded on was that we also localized it in the gut cells, and specifically cells that were not associated with the gut epithelial cells, if I can say this, the gut somatic cells. So we localized it in a cell that was literally looking similar to the undifferentiated progenitor cells. These cells are basically the enteroblasts and the intestinal stem cells. These are cells that literally are important for the regeneration of the gut cells. And if you can see from the highlight, is that this cell that had microsporidia looked somehow similar to the enteroblasts. So meaning that microsporidia in the gut cells is preferring stem-like looking cells. So that can be the reason why also it is hard to horizontally transmit it through feeding and other horizontal transmission or environmental transmission, because it's like it's favoring stem-like cells. So with that, we set also on a journey to investigate how blood meal is affecting this localization and tissue tropism. Reason being that blood meal is important for the insect in that it is changing its physiology, and if the insect physiology is changing upon blood meal, so it's also the microsporidia advancing with the changes. So it's worth noting that there was change of the levels of microsporidia in the gut. The abundance of microsporidia change in the gut upon a blood feeding, so it dropped. Also, it was worth noting that the levels in the gut upon relative quantification was high compared to the absolute quantification. But we can also see that it was not like statistically significant upon checking that. So why is this? Why can this be the cause? We postulate that this can be as a result of high cell turnover after a blood meal. Secondly, we also tried and check on the levels and the levels of microsporidia and their abundance in the ovaries, and we see that there was a slight increase of microsporidia levels in the blood-fed ovaries. Similar results could be shown in in the intensities of microsporidia, both with the relative and the absolute quantification. This can be as a result of the progression of phytogenesis. And when we came to the age effect, the age effect was that microsporidia had high levels in the ovaries compared to the other guts, to the other tissues of the insect specifically. This can be associated also by the fact that the mosquitoes, first of all, were not blood fed, meaning that they were just fed on glucose, and therefore there was like an arrest of the phylogenetic progression, therefore meaning that microsporidia was halted at these points. So with that, the take-home message is that microsporidia is generally in the gonads, and age and blood meal is affecting its level, and also this can be an opportunity for us to explore utilization of this thing, of these facts in terms of improving the intensity of microsporidia, and also looking for appropriate tissues that can be used for further work, which is the omics works and also culturing of microsporidia. Thank you. I thank my team, the SymbioVector team and the CP as a whole, and also my supervisor and the collaborations from Sinking Steel Group. Thank you.
Thank you very much, Edward. I think for the interest of time, you can field the questions in the coffee break. Otherwise, we've got a lot of talks to get through before the coffee break, don't we? So could we bring Joseph up for his talk? Okay, next we have Joseph Chichui. I hope I've pronounced that right.
Yes.
His talk is on, I'm going to read it from there, effects of environmental factors on microsporidia prevalence at the Hero irrigation scheme in Kenya. Joseph.
Thank you. Hello, everyone. I'm going to give this talk on effects of environmental factors and how they affect microsporidia MB prevalence at the Ahero irrigation scheme in Kenya. The Ahero irrigation scheme is located in western part of the country in Kisumu County. Kisumu County borders Lake Victoria, and the scheme is mainly for rice production. Down here we see one of the fields at the scheme. And between July 2021 to December 2020, we embarked on a study where we were collecting Anopheles arabiensis from households in about 9 villages, which I've indicated. I don't think they are so clear on that projector, but what we did is that we correlated this data on microsporidia MB prevalence with environmental data and climate data from the National Irrigation Authority weather station that's located in the scheme, indicated in that that red triangle. So in summary, we collected a total of 63,314 mosquitoes in that period. On this picture, we see one of our team members is called Charles, aspirating mosquitoes from the houses. About 20,814 laid eggs, and in our protocol, we screened those that laid eggs only for Microsporidia MB. 2,266 had the symbiont, translating to a percentage of 10.9 prevalence. On the day that we had the highest prevalence, that figure was 27.202, and the least prevalence we ever got in this period is 1.08%. Also during this period, the daily average temperatures ranged between 18.4. I think something happened. Between 18.4 to 25.4 degrees centigrade. Daily rainfall ranged between 0 to 35.7 millimeters. And the average daily relative humidity was between 35.7 to 90%. So first, to begin with, we want to check how these three parameters interact with Microsporidia MB prevalence. And we start with temperature. What we observed is that whenever the temperature goes down, the prevalence goes up, and vice versa. So also, if I may point your attention to this time between May 2022 to October 2022, when we had the lowest temperatures, that's when we recorded some of the highest prevalence values. And when we did a bivariate correlation, we found that temperature negatively but significantly correlates with Microsporidia MB prevalence. Next, we also did the same for rainfall. And our observation was that whenever there is a rain, there is an immediate increase in prevalence, or the prevalence increases in the subsequent weeks. So I've tried to highlight in blue, it's not so clear there, where we have this kind of trend. And then between January 2022 and March 2022, there was kind of a dry spell. And when the rain came in March, we see that in April, we have a high spike in the prevalence. So our correlation here also says it's positive and significant. Next, we look at relative humidity. And here, generally, the trend of relative humidity coincides with that of Microsporidia MB prevalence. And also, the correlation is significant and positive. So after this, we also want to check how these three parameters relate to the oviposition rates and the indoor mosquito density. And for both temperature negatively correlates significantly with both indoor mosquito density and the oviposition rates. And then rainfall and relative humidity were positively correlated only with oviposition rates. Next, we wanted to check how, since the scheme has a lot of stagnant water for rice growing, what this effect has on Microsporidia MB prevalence. And in these maps, we have a value we have calculated for each month. It's like an index for water cover. So the darkest blue is the highest water coverage, and then the lightest blue is the lowest index. Then here I've only shown for April 2022 all the way to December 2022. And the correlation is also positive and significant. We did the same for bare soil, calculated an index for each month for bare soil, and we find that here the dark brown is the highest bare soil index, and then the yellow one is the medium, and then green is the lowest index. When we did the correlation, we find that Microsporidia MB prevalence is borderline, not so significant, but a kind of negative correlation. So next, we tried to model how these parameters compared to other environmental factors would affect Microsporidia MB prevalence. And we see the water index followed by vegetation index, bare soil, UV index, humidity, dew point temperature, cloud cover. All of them turned out significant with some degree of contribution to Microsporidia MB prevalence. To sum up, is that Microsporidia MB prevalence was highest during the cooler temperatures when we had high rainfall, high relative humidity, as well as when there was more water at the skim and less bare soil at the skim. Mosquito oviposition also responded the same as Microsporidia prevalence. We had high numbers of mosquitoes indoors. I mean, high oviposition when temperatures were low, when there was high rainfall and high relative humidity. Indoor mosquito density was highest in cooler seasons. And then, as we've seen in the previous slide, we have many other environmental factors that influence the host symbiont interaction. So our take-home message is that this Microsporidia MB prevalence in the wild is affected by so many factors, and it will be therefore important to consider this when we are developing a dissemination strategy. Would love to acknowledge our team, SymbioVector team in Mbita, in Nairobi, ANTIVEC, our donors, ISIPE, and the donors of ISIPE, and thank you.
Thank you, Joseph. We do have time for questions, as we're going to have a coffee break directly after this, which I think everybody's pretty happy about. Any questions then for Joseph? Yes, Penny.
Thanks, really interesting. Just wondering if you account for spatial autocorrelation. I might have missed it, but in those statistical models, do you have like a model of the spatial autocorrelation there?
Okay, thank you. We have discussed with data management team at ISIPE, and we want to do that spatial model for it.
Great.
Yes.
Thank you. Coffee time, I think. We'll let Joseph off, and it's time for us all to have a break. So well done, Joseph.
little bit, yeah. Not a problem. Yes. Yeah, because he needs. Okay. So let's change that projector.
Okay, welcome back after the coffee break. We have another talk on microsporidia from Olatane Omitola from the Federal University of Agriculture, Abeokuta in Nigeria.
Hi everyone. I am Olaitan Omitola from the Federal University of Agriculture, Abeokuta. I'll give a talk on a rather young work in progress we are carrying out in Nigeria, the microsporidia species presence and association with insecticide resistance in Anopheles mosquito populations in Ogun State, employing a minimally destructive DNA extraction method. So we've had quite a number of previous talks on microsporidia and microsporidia MB. So I guess we are quite familiar with that already. But briefly, so the microsporidia MB was first identified in the Anopheles arabiensis by Jeremy Herring and colleagues in 2020. And subsequently, it has been detected also in Anopheles funestus in a study also here in Kenya, then in Anopheles gambiae, and as well as Anopheles coluzi in a study by Akole and colleagues in Ghana. All these species are important malaria vectors, and insecticide resistance have also been reported in all species. So why are we interested in insecticide resistance? So it has been shown, it's known that insecticide resistance genotype and exposure to insecticides impacts the development of Plasmodium parasites, as well as other parasites like the filaria worms in Anopheles mosquitoes. And likewise, the exposure to insecticide also is known to impact the microbial composition in a study that was done on Anopheles albuminus by Dada et al. So we are curious to find out how Microsporidia MB and other microsporidians might interact with insecticide resistance in Anopheles mosquitoes. So the broad objective of our study is to determine the association between Microsporidia species presence and insecticide resistance in white-caught Anopheles gambiae and Anopheles funestus populations in Ogun State. So our study depends mainly on DNA extraction using the minimally destructive lysis C buffer. This was originally developed by the Wellcome Sanger team, Mara's group at the Wellcome Sanger Institute, and these are the constituents in a 100 microliter reaction, which we are using as a DNA extraction protocol. Our study design involves collection of white-caught Anopheles larvae from the field and adults, and each individual mosquito will be subjected to DNA extraction as illustrated here. But briefly, where individual mosquito will be placed in wells and submerged in 100 microliter of the lysis C buffer. This will be incubated overnight at 56 degrees Celsius, and the lysate will be diluted at 1:10 ratio. Then we use it for a downstream application in PCR and amplicon sequencing. So we started our study late last year at the onset of the dry season in Nigeria. So between October and November 2022, we collected Anopheles larvae from 8 study sites in Ogun State. We can see them highlighted here in the map. So far, we've screened 380 samples of our Anopheles larvae by pooling and then with qualitative PCR. We screened for Microsporidia MB, and all our samples so far have turned out negative for Microsporidia MB. So afterwards, we also randomly selected 32 of our DNA samples from all the 380 samples and screened them individually this time, and they've also turned out negative. You can see the positive control well amplified there, here. While all the extracted DNA samples, we have no amplification. So what do we plan to do next? So for our next step, we've established so far that our PCR protocol is working fine. So next is to validate our DNA extraction protocol. And with that, we've collected Microsporidia MB positive samples from Jeremy's lab in Isipe. So to verify our DNA extraction protocol and be sure that that is working fine. Then afterwards, we compare the DNA extraction protocol with other extraction protocol like the protein precipitation method that is being used in Jeremy's lab to see how well that works. Then next, we continue to collect more Anopheles larvae and adults from more study sites in Ogun State. We expect the rain to start between March and April this year. Then we resume our collection, and we target screening about 5,000 samples upwards in Ogun State in Nigeria. Then we screen for the presence of Microsporidia MB, and we're also interested in other Microsporidia species besides the MB in our collection. We look at both infection prevalence and infection load in our samples. Then we also correlate this with the presence of Plasmodium species in our white core samples. Then we screen for the cytochrome P450 resistance marker and see how this correlates with between the negative and Microsporidia positive and Microsporidia negative samples. And then we evaluate the vertical transmission of the Microsporidians we detect. And we are also interested in testing the effects of insecticide exposure on Microsporidia MB development and transmission in our Anopheles populations in Nigeria. So I hope to have more interesting findings to present to you in our subsequent meetings. And yeah, so my acknowledgements to these groups at the Federal University of Agriculture, Abeokuta, Osun State University, Nigerian Institute of Medical Research, and then also for the contributions from the Symbio Vector team at ISIPE and the Mara Learning Science Group at Welcome Sanga Institute. Yeah, thank you. Welcome your feedbacks.
Thank you very much, Olatain. PCR negative results for all the lab-based people in this room are very common, so I won't worry about it too much. We haven't got time for questions, unfortunately, so any questions you can field in the lunch break. Okay?
Thank you.
So we have another member of Jeremy's team presenting, and it's Tulu Bukhari. And Tulu is going to talk about community engagement to understand the challenges and opportunities for a Microsporidia-based vector control strategy.
Thank you. Good morning, everyone. I'm Tulu Bukhari, and I'm going to share the results of some community engagement that we did last year. So I had colleagues who had presentations before me. I'm not going to go into detail of all the work that we are doing with Microsporidia MB, and I'm also not going to say much about the study site because that was covered before. But as you've known now that we're at an early stage of our research with Microsporidia MB, but at the same time we understand that for any vector control strategy, and especially one that is considered as novel or as experimental, it is really key that we have built trust with our community for that, and also involve stakeholders at various levels. So we have a very extensive plan of doing that this year, but just to get a feel of it, we did this rapid assessment. So the aim of this rapid assessment was to assess the socio-demographic data, the public health concerns, malaria awareness and management, and also malaria prevention practices, but all of this in relationship to an attitude towards a novel control method that involves mosquito releases. The way we did it is we administered an enumeration and mapping tool with both structured and semi-structured questions to household heads. We trained 40 community health volunteers specifically to use this tool, and in total they interviewed household heads in 44 villages in 2 different locations in Kisumu County in Western Kenya. The quantitative data was analyzed by descriptive statistics, and we used thematic analysis for the qualitative data. So if we look at the socio-demographics, in total we interviewed almost 8,000 household heads, almost equal from both locations. 67% of them were male, 33% were female. 50% were within the age bracket of 30 to 49 years. And 66% were educated up to primary or more than primary level. And the main occupation was farming, followed by casual labor. So when it comes to malaria awareness and management, well, the community recognizes very well malaria is their highest health concern. They know that the vector is mosquito. And they are also very well aware of the core control methods that are there, the bednet, the IRS, and the medication. When we asked them about who they think is at highest risk of malaria, they identified the risk groups perfectly well, but also added elderly into that group. And generally, there was also this perception that everybody is at risk of malaria. When we asked them why they think malaria is prevalent in the region where they live, they attributed it to the stagnant water, which is there because of the rice paddies and rice cultivation that goes on. When we asked them what kind of treatment do they use for the malaria, we found 3 themes that emerged. The first was self-assessment and medication. So whenever they had malaria-like symptoms, there was always this like, am I really feeling that bad to go to the hospital? Maybe I can take a painkiller and see how I feel later. Finance also contributed to it. If there was money, they would go to seek treatment earlier than in the other case. And very few of them actually resorted to traditional medication or to religion. We asked them who makes decision in their household for malaria-like symptoms when there is somebody with malaria-like symptoms. So either it was a joint decision between the parents or it was the decision of the mother. So the mother has a very key role in seeking treatment for malaria. When it comes to malaria prevention practices, we asked them what kind of strategies or prevention strategies they have used in the last 30 days. There was very high use of bed nets, but there was also practices like clearing bushes around the house, mosquito coil, and also drainage of standing water. And when it comes to just bed net, we saw that there was a very high use and also a very regular use by all family members. And then we came to the questions that we were very interested in. So the first question was that, have you ever heard of a malaria control strategy where mosquitoes cannot transmit malaria? And 50% of them said that they have heard of such a strategy. And then we asked them if there is a malaria control strategies where we have mosquitoes that can bite you, but they can't transmit malaria, would you allow us to release these mosquitoes in your compound, in your neighborhood, in the land around your house? And 80% of them actually said yes. And then we asked them, well, if we develop such a technique, would you volunteer with us to implement it? And then the yeses even increased, so we went up to 95%. And initially, when we looked at that data, we said that, well, the yeses have increased because there is always these opportunities that come into a society when there is a project base there. Also, it might be looked at as a job opportunity, but that's not really the case, because when we looked at how we had phrased the question, we had really been specific about compound, land, neighborhood, close to you. So it seems like it's a bit more like I'm okay with it, but as long as it's not close to my house and my family. And when we analyzed the why not, and we looked at the words that came out of it, the words that popped out were like malaria, mosquito, bites. So it is really the pain and discomfort of the bites. But if you look at the female word cloud, you can see another word coming up, and that is compound, so the area around my house. And when we analyzed the data with the socio-demographic data, we found that women were actually less likely to say yes compared to the men. And apart from that, age group, education level, occupation, none of these had any association with the acceptance of this technology. So we conclude that awareness of malaria and malaria prevention is very high. Women have a very important role when it comes to seeking malaria treatment. There is high acceptance for a mosquito release strategy. However, we do need sensitization, especially with women. And 2 limitations of this that I would point out is that, well, the household's opinion, head's opinion, does not represent the opinion of the whole household, actually. And that this community is not completely naive to the idea, because as you've seen from Jeremy's talks and my colleagues' talks before, we have a very heavy presence in this area. We visit them 3 to 4 times a week. But we are considering them in our next phase of community engagement. Thank you very much.
Thank you, Tulu. There's not really time for questions, but you finished your talk ahead of time, which was fantastic. So thank you very much. That'll help a little bit. I'm now going to pass on to the next chair, Damaris. Would you like to sit here? Yeah.
I'll be chairing this session. I'm Damaris Matoke Moher. I work at Cambridge. So our next talk will be from Isaac Asare. Where is Isaac? Yeah, he'll be giving us a talk on spatial temporal dynamics of Burkina Faso, that's West Africa, local strain of Microsporidia and their prevalence with insecticide resistance. You have 8 minutes.
Okay, good morning, everyone. I'm Isiaka Sare from Burkina Faso. It's a pleasure for me to be here to present you our data on Microsporidia as a potential malaria control tool. I know that Jeremy and other speakers already talked a lot on the background of Microsporidia, but I will do like a reminder on the background of this Microsporidia. Microsporidia are known as obligate intracellular parasites, but it can survive outside the host and under the spore form. There is a lot of type of spore regarding the different type of Microsporidia. On this image on the left, you have the spore of Microsporidia. One thing important on this image is the polar tubes. This polar tube is used by Microsporidia to touch the host and inject the sporoplasm in the host. On this group of Microsporidia, we have more than 1,000 species of this group, and some experts on this field think that this is only the fraction of the real diversity of Microsporidia. This means that there is a lot of things to do in this field. Microsporidia have a lot of ways to infect the host. They can infect the host by injection. They can also infect the host by vertical transmission during the contact or the mating. And also there is a horizontal transmission. That's why the female can infect the offspring. Also, it's important to know that this Microsporidia is very specialist. There is around 200 species of this Microsporidia which is very specialist to the insect. Recently, here in Kenya, Jeremy Group found another strain of Microsporidia called MB. This strain have a lot of interesting characteristics that make it suitable for the malaria control. The information of this discovery was the Microsporidia was present in Kenya with different prevalence, and also there are negative correlation between the MB and parasites on the mosquitoes. That they conclude that this could be investigated as a strategy to limit malaria control. Transmission, please. And also, near to Burkina in Ghana, they found the Microsporidia in their samples. Even the prevalence was low, it was around 1.8%, but it was important because it was the first report in this part of Africa. Based on all this information in Burkina, we are trying also to check the presence of Microsporidia in our samples. So we are screening our mosquitoes, we are put in place the positive line of Microsporidia, and we use those lines to conduct some biopsies. I will share with you some of our results. In Burkina, we have principally two main seasons, the dry and the raining season. So what we do, we go in the field, we collect the mosquito for each month, we identify the presence of Microsporidia, and we see and we compare the season, the dry and the raining season. As we can see in this picture, there is Microsporidia all the year. Even the prevalence is not the same, the dry season compared to the raining season. Another thing that we do is because we are running this experience on 3 sites, Gawa, Sumuso, and Vallée du Cou, some person call it also Bama. And we are trying to see the prevalence of Microsporidia on all these sites. As we can see, we have Microsporidia on all the sites, even the prevalence is not the same. There is most of Microsporidia in Sumuso compared to Vallée du Cou and Gawa. After that, what we are interested in is to see the species of mosquitoes regarding the prevalence of the MB, because we have three species, Arabiensis, Clusai, and Gambier, and regarding also the site. What we do, we check the presence of the MB first, and then we check the species of the mosquito where this MB from. And as you can see, unfortunately in Vallée du Cou we didn't get Arabiensis, but in Gawa and Sumuso we have Arabiensis, but the infection of MB on this Arabiensis was low compared to the Gambier and Clusai. After that, we are interested on the resistant strain, on the resistant status of mosquitoes compared to the presence of the MB. What we do, we try to link the presence of MB and the resistant molecular resistance status. We are running the KDR West to see if there is a link between the presence of MB and the resistance status. As we can see, MB is strongly correlates with the molecular resistance status on the mosquitoes. After that, we are also we are interested in the phenotypic resistance. For the phenotypic resistance, what we do, we take the mosquitoes, we put them in contact with the insecticide in the WHO cylinder tubes. We are using as insecticide detamethrin 0.05, and we have two lines of mosquitoes, the resistant and the susceptible, and then we run the MB PCR to see if there is link between the MB and also the phenotypic resistance. As we can see in this picture on the right, there is link between the MB and the phenotypic resistance. As conclusion, I say we should remind that in Burkina we have MB all the year, even the prevalence is not the same. And also the strain of MB that we have in Burkina is strongly correlated with mosquito resistance status, phenotype, and molecular resistance. In the future, what we are planning to do is to check to see exactly on the mosquitoes what is the organ that we have a lot of MB, and also we are trying to see in the field and the swarm if there is possibility to have like infection during the mating in the swarm. And also we will like to check to see if there is co-infection between MB and another useful bacteria like Wolbachia. And also, hopefully, we will get chance to sequence our strain. Thank you so much.
Thanks a lot, Isaac. I think because of time, advice there are no questions, but thanks for sticking to time. So our next speaker is Ewan Parry. He's going to give us a talk on visualizing the life cycle of Microsporidia, a novel agent for blocking Plasmodium transmission. 8 minutes, please.
So my name's Ewan Parry, and I'm working with Professor Stephen Sinkin's group in the CBR in Glasgow in Scotland. And today I'm going to take you over the last year or so of work that I've been doing on visualizing the life cycle of Microsporidium MB, which you know now is a novel agent for blocking Plasmodium transmission. So because of the previous talks, I'm not going to go too much into the background of Microsporidia. I'm going to jump straight into the imaging that we've been doing. But I just want to point out this strain that we've been using is the strain that we've been using is the one that Sari has just mentioned in his previous talk from Burkina Faso. So it's slightly different to the one from Isipe that Jeremy was talking about. So just before I go into the imaging, I'm going to go through our team in Glasgow. We've got Beth Poulton, who's working on the genome, as well as the metabolome of Microsporidia positive mosquitoes, and she's also working on the combination of entomopathogenic fungi and Microsporidium MB. We've got Abdul Hakim Adam, who's our technician, who he's been instrumental in setting up the lines here in Glasgow. And we've got Roland, who's working on the metabolome with Beth, as well as also helping to make the draft genome for Microsporidium MB from Burkina. And Deepak's doing a bit of work on the environmental conditions that affect Microsporidium MB. And he's also looking at some of the transmission from males to females. And then we've got Tom, who's been coordinating the project in general, and has also just been helping out on several different projects. And Cam's the latest member of the team who's just finished his PhD, and he's just starting MB work. So we've got the life cycle of Anopheles gambiae mosquitoes, and I'm just going to go through stage by stage on some of the microscopy that we've found so far over the last year. I'm starting with the eggs. So I've been working with the histology service in Glasgow to develop some processes for sectioning all sorts of life cycle stages from Anopheles mosquitoes. So here we have 3-micron sections of the embryos which have been transferred to glass slides, and then I can stain them how you want. So in these slides I'm going to be showing everything in blue is a nuclear stain, and everything in green is the same fish probe that Jeremy's group has just presented from ICIPE. So this is a typical egg with MB that we see, which has very localized MB signal right at the posterior. And we think this is probably the developing gonad for reasons I'll go into afterwards. We also sometimes see a sort of diffuse signal throughout the embryo in general, which I'm not sure how to explain at the moment. And we also see some embryos with no signal at all. And then finally, we also see some of them have MB positive signals on the outside of the chorion, which they also have their own nucleus. So we think these are probably spores that have been transmitted that way. If we look at the L4 larvae as well, we can section those. And the only place that we see the MB is in these 2 regions at the posterior, which we think are the developing gonad. So in the confocal microscopy, you can see those here in green. And they're right next to the developing Malpighian tubules. And if I zoom into 100 times on the confocal and take Z-stacks, you can see in 3D here that the Microsporidia cluster around the host nuclei, and it's quite clear that they're intracellular. And they each have their own little blue nuclear signal as well. Looking at the adults, this is a bit more similar to what Thomas was presenting from ICIPE. We see them in the testes. So I don't know if you can see on this screen, but the sperm are the long blue streaks in the nuclear stain. And then we have evidence of MB inside amongst them. And we also see them in the duct leading from the testes. And in 3D again, you can see very dense MB signal all around the periphery of the testes and migrating in towards the sperm, but not in the sperm themselves, we think. In the females, you can even see MB at 10 or 20 times on the confocal. So these are subsequent sections. One of them has been stained in H&E and one of them has been stained with a nuclear stain, and the fish probe. And you can see them very densely in the ovarioles, and some ovarioles are much more heavily infected than others, but they're generally there even at low density in all of them. And again, at 100 times you can see this very clearly, and like Thomas, we've seen evidence of ones like this where they're dividing, so it's obviously this is a dividing stage of MB. Interestingly, in some sections you can also cut straight through the spermathecae, so after mating we actually see evidence of positive MB signal in the spermathecae, which is quite nice evidence to go along with what we see with qPCR, that it is being transferred from the males to the females. So that's a spermatheca there cut in half. So the final stage I wanted to talk about was the gravid females. So the gravid females, again, sectioning can be useful because you can see all of the unfertilized eggs sectioned in the abdomen of the female. So here we have a whole female, and if we zoom to this section, you can see MB is quite strongly positive in the remains of the ovary that haven't developed eggs, but it's also migrated into the periphery of the eggs that have developed. And we see it really around the outside of these eggs and towards the posterior pole. And I think this is quite interesting because we're assuming MB is intracellular at this stage, and the only cells that are available in this egg for it to develop in are around the blastoderm, around the outside. And also the pole cells will eventually migrate to the center and generate the germline, which is where we see it later in development. So I think that's consistent with what we've been seeing. So just finally, I want to show a couple of techniques I've been looking into for the future. So some super resolution microscopy I've been able to take from a demo has greatly improved the clarity of what we're seeing at high resolution, and that might help in future experiments looking at MB. And I'm also optimizing some light sheet microscopy, so this is useful for looking at whole tissues, whole mosquitoes, or in this case, dissected ovary. And I'm going to try and optimize this with the fish probe to do the same sort of experiments that we've been doing, but in full 3D. So yeah, that's for the next few months. So over the last year, we've learned a lot more about where MB is in the life cycle. And just to summarize, we found it locally in most embryos. We've localized it to the developing germline in L4 larvae. It's present at high density in both the ovaries and the testes, and we've got visual evidence of it actually in the spermathecae after mating. And then finally, we've found it around the periphery of the unfertilized embryo, so that kind of completes that cycle that we're seeing and where it ends up in the developing gonads later on. So I'd just like to thank Professor Stephen Sinkin's group for all their help, the Histology Research Service for their help with the sectioning protocols, of course Sari and Etienne and Professor Diabate for sending us the samples that allow us to establish the colony in Glasgow, and all of Jeremy's group for letting us use the fish probe and their discussions on the imaging, particularly Thomas, on that side of things. Thanks.
Thanks a lot. I think he has saved us a minute, so we can take at least a question or two. Any questions to?
That's really interesting that you see it in the spermathecae. I didn't quite see that picture, but is it what form is it in?
Yeah, I wish I'd put a bigger picture after I saw Thomas's talk actually. But it's we're seeing both the small forms that you see in the testes as well as the slightly larger elongated forms. So we see both in the few transfers I've seen to the spermathecae.
And you think that you can see spores with fish? Because I mean that's
I don't think we can say necessarily that they're definitively spores until we see polar tubes. I was actually hoping to use a commercial antibody to Nosema, which is a bee-infecting microsporidia. It's the only one I could find, and I tested it on MB recently. Unfortunately, that's not been successful in detecting them. But maybe when we have an antibody, we can say things a bit more conclusively about spores.
Yeah, what's your best model for how coming from sperm it's getting into the fertilized embryo?
It's quite hard to say. I think there's some evidence that it could be as the eggs are passing, it's not actually going with the sperm as it's fertilizing the eggs. It's actually just coating the eggs, because we're literally seeing it just sat on the surface. And I think Jeremy's got some evidence that washing eggs, you get some MB signal from the wash as well. So yeah, it's possibly that, rather than actually going with the sperm and infecting the eggs.
Yeah, thanks a lot for the talk. More questions over lunchtime. So we can go to our next speaker, Lilian Baisi from ICP, who's going to give us a talk on a report on the first draft genome of the obligate intracellular plasmodium blocking symbiont, Microsporidia MB, isolated from Anopheles arabiensis mosquitoes in Kenya.
Hi everyone, my name is Lilian and today I'll be presenting a brief update on the first draft genome of Microsporidia MB. I'm also part of the Symbio-Vector team and I'm doing this work for my PhD at Rhodes University. So without going too much into the introduction, I believe we can all appreciate all the talks that we've had about Microsporidia MB and seeing that it has a very intricate relationship with its host and Plasmodium. So for this talk, I'll be highlighting Microsporidia MB's genetic makeup and also showing some key gene features that we've been able to obtain from the genome assembly that we currently have assembled. So this talk will be three-phased. So we'll start with the nucleotide level annotation, the baseline, highlight some of the key proteins that we've been able to annotate, and also show some of the key metabolic pathways in our assembly. So with the genome of Microsporidia, what we did first was we dissected a couple of highly infected tissues from mosquitoes and sent this out for short paired and read sequencing at BGI. After this sequencing, we were able to get a total of over 200 million reads, 150 base pairs long each. And for these reads, we could assemble, or we managed to assemble a Microsporidia MB genome of size 13.1 megabase pairs. Moreover, the genome that we assembled had a GC percentage of 37.5. We also did a BASCO analysis to identify the core Microsporidia genes from our assembly, and we see that Microsporidia MB has a total of almost 83% single copy core genes based on the BASCO Microsporidia DB database, which contains 600 genes of from 13 referenced genomes of Microsporidia. So with the Microsporidia genomes, they vary in sizes a lot, and some of these sizes, the large sizes of Microsporidia, are attributed to the number of repeats within the genome itself. So we wanted to check how many repeats are in the assembly that we had, and we see that Microsporidia MB has actually a very low level of repeats as compared to some other Microsporidias like Dictyocella, which would have up to 55% repeats in their genome. So having gotten the genome, we wanted to also do a comparative analysis of the content of the genes that we had against other Microsporidia species. So we had around 30 Microsporidia species that we used for this analysis and identified common orthogroups between Microsporidia MB and this reference Microsporidian species. What we see here, I'm sorry it's not very clear, but what we see here is Microsporidia MB actually clusters together with Vitaforma cornea, and this was consistent with other reports where they've solely classified Microsporidia MB using the small subunit rRNA gene. Also, we compared the level of gene content between Microsporidia MB and these other Microsporidias, and what we see is the level of missing genes in red between Microsporidia MB and Vitaforma is pretty much the same. So we did a closer analysis of these common orthogroups between Microsporidia MB and other Microsporidias just to see what proportion of shared orthogroups are within MB and these other Microsporidias. And here we see that the largest proportion of shared orthogroups was between MB and two interesting Microsporidias that have been found in aquatic habitats where we would collect mosquito larvae, and this is highlighted in blue. Also, a high proportion of orthogroups are common between Vitaforma and Microsporidia MB. I've also highlighted here in green several orthogroups shared between Microsporidia MB and other Microsporidias that infect mosquito hosts like Vavrae culissus, which infects Culex mosquitoes, and Edhazardia idis, which infects the Aedes mosquitoes. Finally, we wanted to check the proportions of genes within the functions of the proportions of genes that are in our assembly, and we see that a lot of regulatory genes were found in the assembly, consistent with reports that say a lot of the genes that are conserved over a period of time in Microsporidias are usually useful for membrane trafficking or essential survival of Microsporidia within its host based on its obligate intercellular nature. Then we sought to check if there are any host-exposed proteins within our data set. Host-exposed proteins in Microsporidia are very useful because then they're able to interact directly with the host itself, and also varying from one Microsporidia species to another, you're able to do species-specific gene family sets of this proteins. So from our data set of around 700 proteins of Microsporidia, we saw that we had 8% of these proteins containing signal peptides and 26% of this having membrane spanning regions. However, we're still doing an analysis of the functions of these predicted proteins. Finally, we know that Microsporidia's very key characteristic is that they are spore forming. So we went ahead to check the key structural spore proteins within the data set that we had, and these were polar tube protein, endospore protein, and the spore wall protein. We managed to get a couple of these protein homologs in our data set, but due to time constraints, I'm just going to focus on SWP12. So SWP12 is a chitin binding protein that contains the BAR domain, and we got a homolog that was very closely related to Vita formiconia and had a powers identity ranging between 30% to 80% with other spore wall SWP12 proteins. Moreover, we were able to do a homology modeling of the protein itself from MB, and we could identify the BAR domain within this protein. In conclusion, this just reports the first draft of the MB genome, which stands at 13% and having 83% complete genes that we are using for annotation. I would like to thank my supervisors, Dr. Jeremy Heron and Prof. Tustin Bishop from Rhodes University, and also acknowledge my lab mates who were very influential in the work that was done, and our project funders. Thank you.
Thanks a lot. Thanks a lot, Lillian. I think we can allow 1 or 2 questions because I'm gender biased. It's good to support women in the process. So a question to Lillian. Okay, apparent.
Lillian, I had a question I asked Thomas earlier, but if you eventually can get these genome sequences, would you expect them to be different between the Microsporidia MB that infects Anopheles arabiensis and the one that infects Anopheles gambiae?
I think that's a possibility because Microsporidias are very fast evolving and they'll want to adapt to their specific host. So there might be some variations between different species.
Okay. Oh, sorry, I thought your hand was up. So thanks to Lillian. Let's applaud her one more time. So we are going to take the next speaker, Henry Tonang. Henry is going to give a talk. Here, he's going to give a talk on Microsporidia MB as a bio-based agent for malaria control, insights from mathematical modeling. Welcome, Henry.
Good morning, colleague. And I think I have no much to say about Microsporidia. I will just try to reason a little bit with you because I know my background is totally different from yours. I did physics and engineering, a little bit of mathematics. It means I see things a little bit different. I think it would be good that we try to bring the biology into other science in order to see how we can anticipate, to see how this technology can really be used to enhance or to contribute into the overall goal of eradicating malaria. I mean, what I'm trying to show here is accessing whether spreading in the Microsporidia mosquito population could control the malaria through mathematical modeling. And the first model we did, because we are working in the team, I think towards the end I will show you a little bit the team and the type of contribution we are doing. But here is a very simple illustration. I mean, we took the experiment done through
the vertical and the horizontal transmission of the microsporidia in the normal condition within the lab. And from there, we formulated a very simple model using what is called the SEI approach, susceptible, infected, recover, that has been used for many years into disease epidemiology. And here you have what we call the model assumption. The first thing is you assume that the female mate only once. The second thing is you give a coefficient that represents your vertical transmission and another coefficient that represents your horizontal transmission, and you do the level of attractness. I mean, between the male and the female, you put a certain coefficient, and you also look into the density and the differentiation in terms of the demography between the male and female. And basically, it gives you a certain diagram like what you see there, and you transform this diagram into mathematical equation. And the most important thing is you start doing the analysis of that mathematical equation. You analyze it based on certain set of parameters. What we discover is, in the first thing, there's a possibility of extinction of any mosquito that is positive to mastrosporidia. And what are those conditions? You have some parameters depending on the vertical and the horizontal transmission intensity. You can have that scenario. There's also a possibility of coexistence. What we mean coexistence here is the population of positive and negative mosquito will be leveraging a certain density and oscillating between a certain number within the system. And there's also a possibility, depending on the intensity of vertical transmission or horizontal transmission, there's a possibility that the number of positive mosquito can really go above the number of negative mosquito. But this is when the transmission, the percentage of transmission is very high. But as I've said, when I look into such a system, they are based my analysis on a simple model conducted using lab conditions. But I know our target is there in the field. We are aware that in the field there are what we call weather forcing. And we saw this morning based on the presentation that temperature has a very big effect. And how do we include that variable within our model? Usually, if you go to a location and you take temperature within a day or within a season, it will give you a sort of very fluctuating type of diagram. But mathematically, we take what we call the moving average of that diagram, and we give you a function who can be represented as a sine function with a certain period, as you see in the graph here. And we take this, when we inject this into our model, we start having something very complicated. You start having what we call bifurcation, and depending on your bifurcation, you start having the chaos. I mean, your system does not give you something that you can say for sure that if you do this, this is what is going to happen. And that is really what happened in nature there. And we just got this result based on only one variable. And here we are only studying the population of positive mosquito versus negative mosquito. The real elephant in the room, we have not yet touched it. Because to control malaria, there's another parameter that needs to come in, who is a parasite. They are believed both the parasite and the mastrosporidia live within the mosquito with their host. And I think we are trying our very best to illustrate the different mechanism of interaction. One of the mechanisms may be, I am not sure, maybe if a mosquito grow up from egg stage to adult stage, and before the blood meal, when the
present to the mosquito may parasite the plasmodium. That is one eventuality. Maybe it may change the structure of the host and not allow the parasite to complete the cycle. That is another eventuality. There are several eventualities, and we are trying to introduce all this type of eventuality into a more complex system of modeling by using simulation. In collaboration with the Millennium Institute, we are using what is called the system dynamic approach. With this mathematical model, we're able to know the area of convergence or a parameter. Now we can boldly introduce this into a system dynamic. I think Jeremy showed you a little bit the slide of very complex and do a lot of simulation, do what we call scenario analysis and intervention analysis in order to see how this can happen. The second thing, we are also working with the agent-based model in collaboration with Prasan, who is down there, and see how we can combine this, because at the end of the day, we would like to be sure that this methodology can work. But to conclude, I would like to tell you all models are wrong. Yes, I am sure of that. But they are very useful, and they can really do something that even your analysis may not help you to do in the lab. Thank you very much.
Thanks a lot, Henry. All models are wrong, but they are very useful. I wonder whether we should go that way. So I think we have a few minutes. I'm told there's been a few adjustments to the program. So we have a few minutes for questions for him and the previous speakers. Any questions to Henry? You agree with the statements that all models are wrong? Okay, a quick question to you. Do you take care of the other confounding factors? Do they play a role? Do they influence how you present the model, such as environmental factors, etc.?
What I presented, I initially showed the model development under optimum condition. Now, I took the same model, I took only one variable was temperature. And based on that, I was able to tell you how there's changes. That means you reach a point where the model can no more help you to predict. Now it starts behaving a little bit strange. When it gives you what we call the butterfly mechanism, it means you are in the area of chaos and your model is no more applicable within that boundary. But now if you are to inject rainfall or relative humidity,
yes,
I'm sure you increase the complexity. But nevertheless, sometimes when you use simple mathematical model, it's a bit complicated. And that is why we are also collaborating to bring some different methodology who are more simpler to integrate as many parameters as possible. That's why we are using system dynamic. At the same time, we are using agent-based. But some of this work has been presented under poster with the 2 PhD students that we have within the team. And they have more detail about some of the results here. I just give the bigger picture of what we are doing, but some of them have zoomed into some of the examples where they pinpoint some environmental variable like even land use, the NTVR that show the vegetation, the number of water, pool, and all these things. Thank you.
Yeah, thanks a lot. We have 5 minutes. For the previous speakers, any question, such as Isaac? Do we have any questions for him? Okay, I think we are good. Oh, Jeremy has a question.
So just on the insecticide resistance phenotype that you saw, so if I'm correct, the microsporidia infected mosquitoes were more susceptible. Is that right? And do you have any ideas what like the mechanism for that could be?
Sorry, I didn't get you very well. The question.
Hi, so I'm Steph Rainey from the CVR in Glasgow, and I'm the chair for the next session. So we're running a wee bit behind, so we're going to try and fit everyone in before 12:30, but if not, we'll go for lunch at 12:30, and then anyone that's not spoken can speak after lunch. So we're going to start this session with George Demopoulos, and he's going to talk to us about targeting pathogens in mosquitoes.
Okay, well, thank you. I would like to thank the organizers of the meeting to invite me. It's really exciting to be here and meet old friends and make new friends and forge new collaborations, perhaps. I'm going to talk to you about mosquito pathogen interactions and how we can block these interactions with a major focus on malaria. And I have introduced several alternative stop codons in my presentation, so we'll see how far we can get. So this talk is very much focused on a population replacement strategy based on gene drive. And we don't work on gene drive. We collaborate with a consortium of laboratories, the University of California Malaria Initiative with Tony James, Ethan Bayer, Greg Lanzaro, and John Marshall. And my lab is specifically focusing on blocking the pathogen in the mosquito using genetic engineering. That's what I'm going to talk to you about. So when the malaria parasite is infecting the mosquito, it engages in a number of different interactions with so-called host factors. These are mosquito-encoded proteins that the parasite has to interact with to complete its infection cycle. At the same time, it's attacked by the mosquito's innate immune system that comprises a variety of anti-parasitic restriction factors. And we can use host factors and restriction factors to render mosquitoes less permissive to infection. So for example, we can remove host factors using genome editing or nanobodies that interfere with those interactions, or we can add and overexpress restriction factors. Restriction factors fall into two broad categories. We have the endogenous restriction factors. These are mosquito-encoded immune factors, most of them. We also have exogenous restriction factors. For example, the melittin antimicrobial peptide that Nikolai presented yesterday is an example of such. These are non-mosquito encoded factors. And when it comes to host factors, we can either genetically disrupt them using CRISPR-Cas9 genome editing, or we can mechanically disrupt those interactions using nanobodies. I'll show you examples of that. So the malaria parasite has a complex infection cycle starting with the ingestion of gametocytes throughout the stage of sporozoite injection into the host. And I'm going to walk you through each one of these stages and show you examples of how we can render mosquitoes less permissive to the parasite genetically, exploiting these different developmental stages of the parasite. So the first stage is ingestion of the gametocytes. This is really the blood feeding stage. And many of you may think that, well, this stage is probably not very relevant for finding a way to block the mosquito, but it actually is. It turns out that during blood feeding, the mosquito is injecting saliva into the bite site and at the same time ingesting blood, with the result of that saliva becoming blended into the blood meal. And some very recent and exciting research coming out of the NIH has shown that the saliva contains a variety of factors that are really important for blood processing and digestion, as well as parasite infection. And an example of that is a mosquito-encoded protein called GILT, which we studied several years ago in collaboration with Errol Fickrig's lab at Yale. And GILT is a salivary gland-specific protein that is injected into the wound site. And we showed back then that it associates with a sporozoite, where it modulates the efficiency of infection. More recently, we created a knockout mosquito for the GILT1, so a mosquito that does not have GILT1, and we looked at how efficient the parasite could infect those mosquitoes. And surprisingly, we noticed that the parasite had a harder time establishing an infection in the mosquito when GILT was not there. And the only explanation for that is that GILT is in some way ending up in the midgut lumen, where the parasite is performing its first stages of infection, and it's facilitating infection. And when you remove it, the mosquito becomes less permissive to infection. So you can drive anti-parasitic effectors using salivary gland promoters. And the advantage of that is that you can target very early stages of parasite development. If you use the regular blood-feeding inducible promoters, like the carboxypeptidase promoter, you will not be able to target those very early stages because they happen in the first few hours. Here is another example of that. This is experiments where we used a nanobody that is targeting a host factor in the mosquito. And what we realized early on, when we fed mosquitoes on a gametocyte culture to which this nanobody had been added, we saw a very significant reduction in infection, as you can see on your left panel. And when we added the nanobody to a gametocyte culture of Plasmodium falciparum, we saw no exflagellation. So this nanobody is also interfering with a parasite development at the stage of gametocytogenesis and exflagellation. And the only way you could actually target the parasite, we believe, with this nanobody in a transgenic fashion would be to express it in the saliva. And nanobodies, for those of you who don't know, are the variable region of cameloid antibodies. They have a very high level of specificity similar to that of human antibodies, but they can be encoded by a single small gene. The next stages of the parasite occur in the lumen within the blood meal. So we have the formation of the zygote and the motaleokinete. And within that blood meal, the parasite can actually be targeted. And we showed that several years ago when we manipulated mosquitoes in a way that they would have a stronger immune response to the parasite using the IMD pathway. And what we saw here was that we saw a smaller number of okinetes in the mosquito lumen when you activated the IMD pathway using blood meal inducible REL2 transcription factor. So the parasite can be targeted within the lumen. The next stage is invasion of the midgut epithelium, and that is preceded by traversal of the peritrophic matrix, which is a chitinous layer that lines the midgut epithelium from the inside. It compartmentalizes the digestion of the blood. And this is perhaps the stage with the greatest vulnerability of the parasite to the mosquito's immune system. Most of the mosquito anti-plasmodium immune responses occur at this stage when the parasite is invading the midgut epithelium. So that is an advantage of targeting that stage because we actually know about many antiparasitic effectors that are acting at that specific stage against the parasite. We also have blood meal inducible promoters, which we can drive expression of those effectors. A disadvantage of targeting the parasite at this stage could be that the parasite is a moving target and it's a short time window, a relatively short time window, about 10 hours. You got 10 hours to block the parasite here. I'm going to show you examples of effector systems that can block the parasite in the midgut epithelium. The IMD pathway, one example. If you overexpress the REL2 NF-kappa beta transcription factor in the gut of the mosquito after a blood meal, you will see a very significant blocking of infection. And you can see those green dots there is a number of oocysts you see on the guts of different mosquitoes that express the REL2 in the gut or in the fat body or in both tissues. You can also manipulate the IMD pathway in other ways. You can overexpress other pathway components, or you can overexpress downstream effectors of that immune signaling pathway to render mosquitoes less permissive to infection. Here is an example of a transgene that produces 5 different antiparasitic factors. Some of them are endogenous and some of them are exogenous. Scorpion is an example of an exogenous antimicrobial peptide, as is VIDA3. And when you express this construct in the mosquito gut after a blood meal, after ingestion of a gametocyte culture, the parasite will get targeted both in the midgut epithelium and in the lumen, and with a very significant suppression of infection. So here is now an example of a mosquito host factor. This is C-type lectin-4. It protects the parasite from melanization. Early studies showed that if you silence CTL4 with RNAi, you would see a prominent melanization of the O-kinates that are invading the midgut epithelium. We developed a CTL4 knockout mosquito a few years ago, and that would very effectively melanize Plasmodium falciparum parasites as they are traversing the midgut epithelium, with a 64% decrease in infection prevalence, which is highly significant. And you also see on the very right panel that the number of sporozoites in the mosquito salivary gland also very significantly decreased. Another host factor is FREP1. FREP1 stands for fibrinogen-related protein 1. It's a protein that forms a tetramer and is associated to the peritrophic matrix. The parasite has to interact with FREP1 to get through the peritrophic matrix. And if you remove FREP1 completely using genome editing, the mosquitoes become highly resistant to parasite infection, as you can see in that. I wish I had a laser pointer, but if you can see in that panel with those dots towards the left. And the interesting thing with FREP1 is that some genome association studies identified 2 alleles of FREP1 that differ by 1 amino acid. And those studies suggested that the allele that has a leucine is function as a host factor, while the allele that has a glutamine doesn't function as a host factor. Okay? So what our collaborator Ethan Beyer did at UCSD, he developed an allele replacement mutant. So a mosquito where he replaced the host factor functioning allele of FREP1 with the one that supposedly doesn't work as a host factor. And you can see here that there is again a very significant suppression of infection when it has the glutamine amino acid allele. You can actually make a replacement mutant for FREP1 by changing one single nucleotide. Leucine to glutamine, one single nucleotide change, which becomes kind of an interesting application in a gene drive perspective. You just have to change one nucleotide and you can get the mosquito that is highly refractory to the mosquito, and something we are working on with Ethan's lab. Here is a final host factor, which we identify through serendipity. It's a prefoldin chaperoning complex. There are 6 prefoldins encoded by Anopheles gambia. Prefoldin interacts with a chaperonin TCP complex, and it's involved in microfilament and microtubule formation. And the interesting thing is that if you silence any of those 6 prefoldins, mosquitoes become more resistant to infection. And one thing that really inspired us with this prefoldin complex is that you have so many different targets that you can now inactivate and yield a refractory phenotype. It works in Anopheles gambia, Anopheles stevensi, and Anopheles dirus with Plasmodium falciparum, Plasmodium berghei, and Plasmodium vivax. That's why we call this a pan-Plasmodium host factor. If you immunize a mouse with prefoldin, you can see transmission blocking activity of those mice. So it can also work as a transmission blocking vaccine target. Let's move over to this stage here. This is the stage where the oocyst is developing. And this is probably the least vulnerable stage of the parasite to the mosquito's immune system. But it provides a very long time window of opportunity for targeting. The oocyst is going to stay there for about 10 days. It's sitting there. However, one problem with targeting this stage is that there are barely any, if any, known mosquito immune factors that can target the oocyst stage. The oocyst seems to be camouflaged, but if you could target it, you have a very long period by which you could, attack it. And during that 10-day period, Anopheles mosquitoes in the field, they're going to acquire several blood meals. And that's important because the promoter that you will drive your effector system is going to turn on multiple times during that time period. So Johnny in the lab, a postdoc, he used to work on the melanization system of Anopheles gambiae. So he has a very good knowledge of different serum proteases and cliptomane serum protease homologs that regulate melanization reactions. And through a screening, he actually identified 2 serum proteases that, when silenced, can achieve melanization of late oocysts on the mosquito gut. And these are very preliminary data. The blocking is not complete, is far from complete, but this is also a silencing experiment where you can never really deplete your target protein completely. We don't even know how much of the protein we are depleting in this case. But we are very excited about this finding because in theory, if you could inactivate both or one of these cliptomane serum protease homologs, you might actually achieve blocking at the oocyst stage, and it gives you that very long time window. The next stage is the stage where the sporozoites are released from the oocyst and they migrate to the salivary gland. Well, they don't really migrate to the salivary gland. They will get distributed throughout the mosquito hemolymph, and randomly some of them will end up in the salivary gland, about 20%. This is a relatively short time window of opportunity, and we don't really know much about the mosquito's immune response against that stage. I'm actually not aware of many immune factors that could be utilized to target that stage, but there are exogenous antiparasitic effectors that one can use. And an example of such is a nanobody fusion, a CSP nanobody fusion with a scorpion. Scorpion is a scorpion toxin, and the CSP nanobody can bind to the circumsporozoite protein that is coating the entire surface of the sporozoite. So what it does, it brings the scorpion onto the sporozoite. And with this construct expressed in the fat body of mosquitoes, we can achieve a very significant blocking at the sporozoite stage. So this is an exogenous antiparasitic effector. The sporozoites will from there end up in the salivary gland. And I am actually not aware of any effector system that has been studied or identified that can target the sporozoite in the gland. And maybe someone knows about the system. I am not aware of any. It would be very interesting to be able to target it there because also there you probably have a pretty large window of opportunity. The final stage is the injection of the sporozoite into the human host. And it turns out that the saliva actually contains a number of proteins that can modulate this transmission and the efficiency of the sporozoite to infect the host. The first example is SAMS1, which enhances infection of sporozoites both in vitro and in vivo. The second example is gilt that I showed you earlier, which is coating the surface of the sporozoite and makes it more difficult for it to infect. And the third example is trio, which is required for liver infection of mice with Plasmodium berghei. And when you knock out trio, you actually see a significant loss of infection in the mouse liver. So you can actually express or remove factors from the mosquito saliva and influence transmission to the human host and infection in the human host. So there are multiple effector systems that one can use at all of these different stages. And that's a good thing. The question is, which one of these can actually meet GM mosquito target product profiles? And we don't have time to go into all those details. But it might be a good thing to have multiple effector systems to mitigate the emergence of the resistance. For example, by making transgenic mosquitoes that can express multiple factors. Or when you release a mosquito that is blocking the parasite might develop resistance to that blocking mechanism over time. It might be good to have the next set of effectors to spread. So that's why we like the idea of having multiple factors. Okay, I'm going to show you one little story now on arboviruses to Aedes aegypti. It's only be 2 minutes, but it's an interesting concept. Okay, you could probably apply it for Anopheles too. When it comes to targeting arboviruses in Aedes aegypti, we have exploited the siRNA pathway, small interference RNA pathway, because it's a pan-antiviral defense system. It's active against all RNA viruses, hence all human pathogens that are transmitted by mosquitoes. And what we have done here is that we have overexpressed some of the key factors of the siRNA pathway, dicer2, R2D2, and Argonaut2, to create mosquitoes that have an augmented siRNA pathway. And on your left panel, you see overexpression of dicer alone, which results in a significant decrease in infection prevalence, as well as infection intensity. In your right panel, you see an experiment where we overexpress the 3 factors together. And here you see a highly significant decrease in infection prevalence in the mosquito population. So you can augment the RNAi pathway and achieve transmission blocking. Another interesting thing you can do with the siRNA pathway is to inactivate it. If you inactivate it by either knocking out Argonaut2 in your left panel there or dicer2 in your right panel, you make the mosquitoes significantly more susceptible to virus infection. And the nice thing with that is that the mosquitoes die of infection. So here, for example, you can see the mortality of mosquitoes in Argonaut2 knockout mosquitoes. And in some cases, with some viruses, Mayarovirus, you have a very significant mortality. And the same thing happens when you knock out dicer2. You see a very significant and increased mortality of mosquitoes compared to the control. And a dead mosquito is a good mosquito. So in theory, if you could inactivate the siRNA pathway at the stage, in a stage-specific manner, that mosquito, when it gets infected with one of these human pathogens, should succumb to infection. And with that, I'm going to close. And we might, I don't know if we have time for maybe a question or two. Thank you very much. Well, before I go there, I would like to thank the different people from my group that have worked on these projects over the years, our collaborators, and obviously our funding agencies. Thank you.
Thank you, George. Yeah, I think we've got time for maybe one question.
Yeah, really interesting stuff. I'm just wondering about that incomplete penetrance of blocking in the oocyst. Do you see that some oocysts are not melanized, or some of them are incompletely melanized and you get some escaping sporozoites from those sort of?
No, that's a very good question. So there is there's some oocysts that do not seem to be melanized at all. Some oocysts are strongly melanized, and you also see some partially melanized oocysts. We don't really know how that translates into sporozoite infection yet. These are very preliminary data. And we think that the incomplete melanization that you see is probably a reflection of using RNAi. It's something we have seen in the past too. It's very difficult to deplete the serum proteases completely. But the significance of that finding is that you can melanize oocysts. You can actually manipulate mosquitoes to melanize oocysts. You just have to find a way to augment that response.
Thanks for the lovely presentation. Can you comment on the relative fitness costs of the various approaches in Anopheles and also for the AD's approach potentially inactivating sRNA, given ISVs circulating, would there be pretty high fitness costs there?
Absolutely, that's a very good question. The fitness cost, and that's really part of that target product profile I was mentioning at some point. We have all these different effector systems, and obviously some of them would not work. Some of them have such severe fitness costs that the mosquito barely makes it to the adult stage. Like CTL4 knockouts, for example, pre-folding knockouts would not work. But what those knockout models are showing us is that if we can find an alternative way to inactivate those factors, we can achieve that highly resistant phenotype. FREP1 is a good example of how we found an alternative way to inactivate host factor function of that protein without any fitness cost, because it can still carry out its mosquito-specific function. In general, knocking out genes always going to have a fitness cost. Overexpressing genes does many times not have a significant fitness cost, because the overexpression is usually transient through these blood meal inducible promoters, not on all the time. Thank you.
Have you explored quorum sensing signals? The impact of quorum sensing signals to see whether that affects the number of oocysts or their size.
Quorum sensing signals?
Yes.
We haven't looked at that.
Okay.
Thanks, that's great. Thank you. So our next speaker is Howard Traore from the IRSS in Burkina Faso, and they're going to talk about blocking Plasmodium development in mosquitoes using Serratia.
Good morning, everyone. My name is Awa Traore, and I'm a PhD student in molecular biology at the Université Nancy Boni in Burkina Faso. My talk will be on blocking Plasmodium development in mosquitoes using bacteria Serratia euritica YN1. My presentation will follow this plan. In introduction, malaria is one of the world leading causes of death, particularly in sub-Saharan Africa. Children and pregnant women bear the greatest disease burden of disease. According to the WHO, Plasmodium falciparum causes more than 95% of malaria cases and 96% of deaths. Its main vector or Anopheles gambiae. Current malaria control strategies such as the use anti-malaria drugs and vector control have reduced disease burden in many endemic areas. But the emergence and rapid spread of insecticide and drug resistance are limiting these strategies. Therefore, malaria eradication is needed with introduction of new control techniques in addition to traditional control measures. Novel ideas for control of mosquitoes-borne disease include using bacteria symbiotes such as Wolbachia, Asaya, and Serratia. We are exploring the feasibility of development in alternative strategy to fight malaria by using Serratia euritica YN1 that live in mosquitoes good to suppress Plasmodium development. The general objective of our study was to investigate the ability of Serratia euritica YN1 to prevent Plasmodium development in mosquitoes. And specific objectives were to determine Serratia SPP prevalence and to assess the impact of SUN's infection on mosquito survival, and finally to determine SUN's ability to inhibit oocyst prevalence and intensity. To obtain our result, we first collected mosquitoes in locality of Bama and Sumuso. and determine the presence of Serratia spp by conventional PCR by targeting laxus gene. And band size of Serratia was 150 pb predict. For infection of mosquitoes was with bacterium, we first isolated Sunds on LB plus ampicillin medium, and Sunds colonies were used to prepare a suspension bacterium, and the suspension bacterium was applied to the stored cotton balls and placed on section containing the female anopheles. Then to evaluate the effect of Sun on mosquito survival, the dead mosquitoes were removed and counted, and we used PCR to verify the presence of the Sun. Bacteria infected and infective mosquitoes were fed a blood meat containing gametocyte, and stomach mosquitoes were dissected in their sterile condition, and we use microscopy for determine the presence of oocysts. In result, there were 456 mosquitoes were collected, with 187 being larvae and 269 being adults. And the overall prevalence of Serratia was 12.28. And in this study, Serratia did not have an effect on mosquitoes' survival. Gao and Odd found the similar result. This result explains that Sun is a symbiote of mosquitoes, and symbiote contribute to mosquitoes' physiologic well-being. In our investigation, Sun showed a lowering effect on prevalence and intensity of oocysts. Indeed, the prevalence and intensity of oocysts were lower in treated mosquitoes than in control. And with 55.36% oocyst reduction, this result explains that Sun secret amplit is a lipase that destroys plasmodium membrane by destroying a lipid that a major component. In our study, Sun's effect was significantly higher carrier B than carrier A. This result suggests Serratia inhibitory effect of the bacterium on oocyst intensity was dependent on the parasite density of carrier. So in conclusion, this study found Sun's strength have a reducing effect on the prevalence and intensity of oocysts in mosquitoes, supporting inhibitory effect on plasmodium development. As a result, this bacterium could be a potential tool for blocking malaria transmission in fields. In perspective, we will want to study approaches for the dissimulation of Sun under semi-natural conditions and to evaluate its effect on non-target organisms and persistence in the environment. We end our presentation by thanking all people who took part in this study. Thank you for your attention.
Thank you very much, and thanks for keeping to time. So we've got time for a question. So we've got a question down the back.
Yeah. Thanks for your nice presentation. My first question is, which Anopheles species did you use in your experiment? The second one is, for the mosquitoes infection, were you sure of the absence of the other bacteria? Thanks.
Thank you for your question. And we 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 use Anopheles gambiae for your study. Anopheles gambiae. And to and we was isolated since Ceresia on medium contained and medium selective contained antibiotic ampicillin that enable the development and of bacteria.
We'll move on to the next speaker. Thank you. So our next speaker is Ife de Mores from Yale, and she is going to talk about anti-plasmodium bacteria and looking in the microbiome of Anopheles.
All right. Hi, everyone. My name is Ife Desamour. I'm a PhD candidate at Yale University. And today I have the pleasure of sharing this part of my thesis project with you, where I am focusing on identifying novel antiplasmodial bacteria in the Anopheles microbiome. So a little background to start. The mosquito microbiome plays a role in mosquito development, nutrition, reproduction, insecticide resistance, and today's focus, susceptibility to pathogens. Here we have a simplified diagram of the mosquito anatomy, where you can see the organs that are highlighted are all known to be colonized by bacteria. And the reason that we're interested in this is that not only are these colonized by bacteria, but a lot of these organs are sites of plasmodium development. So we're interested in these for a novel vector control method. And so research conducted by some of the people in this very room has shown that the Anopheles microbiome protects its host from plasmodium infection. Specific bacteria have been shown to inhibit plasmodium development by either activating the immune response or secreting antiparasitic factors. And so my overall question is, can we identify some novel bacterial symbionts with antiplasmodial activity? And I asked this one because I think it would be really interesting, but also because the bacteria that have been discovered so far have only been discovered in a handful of countries, and even from some lab-reared mosquitoes. So given that the mosquito microbiome is environmentally acquired, we've only begun to scratch the surface on bacterial diversity, and by increasing the number of geographic locations surveyed, we can reveal some novel symbionts. So to address this, I'm starting by comparing the microbiomes of infected and uninfected blood-fed field-derived Anopheles mosquitoes. And so I hypothesized that Anopheles mosquitoes that are naturally infected with plasmodium have a lower abundance of bacteria in their microbiomes than uninfected mosquitoes. And so here we have a graphical representation of my hypothesis, where this blue bacteria is our hypothetical inhibitory symbiont. You can see it's at a much lower abundance in this infected mosquito, whereas here it's 7 times more abundant in this uninfected mosquito. And so you may be wondering why the focus on blood-fed mosquitoes. So first of all, the gut microbiome population expands after a blood meal, so there's a much higher abundance of bacteria at this time. Second, due to this proliferation, there is actually a widely antimicrobial immune response that is triggered, which can impact the parasite. This is also a key point where the bacteria and parasite share a space, and are able to interact with each other since they are then physically separated after the O kinase cross the peritrophic matrix. And so the crucialness of this point is also further exemplified by the fact that symbionts with known mechanisms of inhibition all affect O kinase. So we may be able to enrich for inhibitory bacteria by looking at blood-fed mosquitoes. And lastly, there is a knowledge gap for blood-fed mosquitoes. So field studies to date have all focused on the microbiomes of unfed mosquitoes. And so to address this, we went to Senegal, a country where no inhibitory bacteria have previously been isolated, and we specifically traveled to this region over here called Kédougou, which has a high burden of malaria in order to enhance our likelihood of collecting infected Anopheles. And so although this region only accounts for 1.2% of the Senegalese population, it accounts for 19.7% of malaria cases, so it has a very high burden. So we conducted household aspirations in 13 villages and collected 493 blood-fed Anopheles mosquitoes. Most of these were, as expected, the main malaria vectors of the region, so Anopheles phonestis and Anopheles gambi. However, 14% of our samples were Anopheles rufipes, which I've labeled as an emerging malaria vector purely because only a few studies to date have reported having infected Anopheles rufipes. And so recent studies have shown a behavioral change where it is becoming more anthropophilic and also being more commonly found indoors, as corroborated by our study. And so, unfortunately, I don't have much data to share yet, as my samples only arrived to the US very recently. But so I'm currently generating 16S rRNA amplicons of individual mosquito abdomens to characterize their microbiomes. So this will be able to include both the midgut and reproductive tissues. I'm also detecting plasmodium in individual mosquitoes, separating the head thorax from the abdomen in order to differentiate the midgut stages from sporozoites. And then once all that data will be gathered, I'll be able to do some bioinformatic analyses to do differential abundance work and compare the microbiomes of infected and uninfected mosquitoes. And so for part two, my second aim is actually to identify these novel bacteria that inhibit Plasmodium falciparum development specifically. So from those same blood-fed mosquito samples, we isolated culturable bacteria from the mosquito abdomens. And so we then analyzed colonies using the MALDI-TOF biotyper, which basically uses mass spectrometry to measure protein abundances. And it matches these protein patterns to a really extensive reference library in order to be able to identify different bacterial species. And so from 94 different mosquitoes, we identified a total of 69 different bacterial species. And so I will be screening these for antiplasmodial activity, first by focusing on those that are correlated with uninfected mosquitoes from my first aim, also looking at genera with known antiplasmodial activity, and then looking at genera with activity against other vector-borne diseases. And so we tested 280 colonies. 32 of these colonies remain unidentified. As I said before, we have 69 different bacterial species, and of those, 6 are from genera with known antiplasmodial activity. 3 are specifically species that are known to have antiplasmodial activity, and then 3 are from a genus with activity against other vector-borne diseases. So Licinus bacillus decreases Zika infection and also decreases leishmania infection in sandflies. And so next, I will be Sanger sequencing these unidentified bacterial colonies. It's possible that they were simply not in the MALDI database, but there's also a very small chance that perhaps we have something new, which could be very interesting. And then I'll be screening these for antiplasmodial activity by colonizing my mosquitoes by sugar, which was shown by Hawa in her presentation, and then followed by the standard membrane feeding assay to look at the antiplasmodial activity. And so this was a huge collaborative effort with lots of people. Special thanks to my PI, Amy Bay, and to our team at Pasteur that helped out with this. And with that, maybe I can take questions. If not, here's my email.
Thank you. Thanks for another great talk. I think we've got time for one question. Okay.
Thank you very much. Nice talk. Is there any reason to focus more on bacteria and not other organisms?
Personally, there's a lot of interesting stuff in the microbiome, definitely. I honestly just chose bacteria because I'm really interested in bacteria. No other specific reason.
Right. Thanks. If there's any more questions, we can maybe do it later on or email, which is great. Thank you. Thanks very much. So our next speaker is Martin Rono from Emory Wellcome Trust Research Programme here in Kenya. And it's a talk regarding vectorial capacity and TP1 genotypes of Anopheles gambiae sensilato mosquitoes on the Kenyan coast. Thanks.
Okay, thank you very much. First, I have to thank George for giving a nice overview, so I don't have to give an introduction to mosquito immunity and the vectors. But microphone. All right, I hope you can hear me. Thanks. I said just first I want to thank George for a nice presentation of the different system in mosquito that can control parasite infection. I'll dive down to one called TEP1, which is Saisa containing protein 1. And we are looking at the mosquito population within the coastal region of Kenya. So my work was informed from this paper that I contributed part of it. And they were looking at how mosquitoes have co-evolved with the parasites and how they're structured within different regions. And what was interesting is to find a mosquito population called Anopheles maris that is locally found in Kenya that has one genotype that was unique for the TEP1 refractory allele, so called TEP1R3. So I wanted to understand more about this genotype and the mosquitoes in view of malaria transmission and as probably a tool for controlling of infection, maybe using gene drive. So the importance of the R alleles. So here in the representation here, you have the number of mosquitoes that are refractory to infection with the type of genotype. And you can see R at the extreme end and S, which is susceptible. So the susceptible normally allow parasites to develop that, though they still control to some level. So the R3 is one of the refractory allele that was discovered in the coastal region. So I wanted to characterize that mosquito population. And below you can see parasite numbers. When you have R, the dark blue, you have very limited number of parasite developing. And when you have the S, you have high parasite populations. And here, I think in Robert Sauer and group, they showed that even in falciparum, previously it was work on bug-eye, but even in falciparum, if you inactivate the TEP1, you see higher parasite development, meaning that it might be an important factor in controlling infection in mosquitoes. So we went about looking at the mosquito that have the TEP1R3 genotype in one region of not far from here in Malindi. So it's one region that we found most of the samples that we previously seen in the publication, and it is near the ocean. The ocean is on the left, and these are the houses that we sampled the mosquitoes. This was done over 6 days, and we use standard approach, CDC light trap, then you do microscopy determination of the type of mosquitoes, and then you look at different parameters from the mosquito's head, thorax, and the abdomen. And these are some of the techniques we employ. So what was interesting, after 6 days of sampling, we had almost 20,000 mosquitoes. There's high level of
mosquitoes moving around. Most of them were Culex as usual, and then you have some Finestus and Gambiasis and some minor species. Yeah. And then you looked at in-depth in what kind of sibling species are in the Finestus group, and those are the ones indicated the left side. And for most important was the one in Anthopheles gambi. We only found Arabiensis and Meras. So we didn't find any SS in this region. And also from other studies, we haven't been able to capture SS. So there's been a bit of changes. So maybe even techniques that are targeting Anthopheles gambi SS might not probably work in this region, might need to rethink the whole process. And then when you look at the behavior of the mosquitoes, majority of the mosquitoes were outdoor. So they feed outdoors, they rest outdoors, but a few of them indoors. And then you look at the parasite numbers, majority of the outdoor is where we found positive, and for Anopheles meras, which is, I hope I can figure out how this one works. Can't see the point. Yeah, so the high numbers were outdoor for Anopheles meras, meaning that they are important vectors for transmission, and they are becoming more increasing, and they are more adapted to the salt breeding regions at the coast here. And then you looked at the type 1 genotype. On the left, Anopheles arabiensis, on the right, meras. And just to orientate you, the orange is the refractory allele, which is more dominant in arabiensis, and the blue and the purple are the susceptible ones, which are seeming more in the meras. This is just how type 1 kills. The S type normally leaves a few mosquitoes parasite developing, and they are normally killed by melanation and lysis. So we think in meras we have more potential to transmit malaria compared to the arabiensis. So in conclusion, we found lots of Anopheles meras and arabiensis. We didn't find any Anopheles gambi SS. They're mostly exophilic. They're relaxed feeders. They feed on animals and humans, so they can escape some of the control measures. So we need to rethink about that. And we have a lot of allele types in Anopheles meras, and majority of them are the susceptible one, but of interest was the refractory allele, three, which we don't know if it's working as the other refractory allele. So we are trying to design experiments now to test for infection status in that genotype. And lastly, the high prevalence of Plasmodium infection in Anopheles meras suppose that we need to rethink about the species that were initially looked at, minor species that might be also contributing to infection of malaria, and we might need to think about control tools for them. The outdoor feeding, the relaxed feeders, and they are initially not targeted for control. Yeah. This work was done with my research assistant there, Brian, and colleagues who assisted in different ways, and the funding scheme that provided the funding. Yeah, welcome.
Thank you. We've got time for some questions. Yeah, so thank you very much. Thank you. So we're going to head to lunch now. I'm sorry to the speakers that thought they were going to be able to relax over lunch. One more? One more? Okay, one more. We're going to do one more. So okay, so last speaker before lunch then is Claudine Taskenko. Sorry if that pronunciation is not right. And they're going to talk about effects of Isaiah species on the fitness of Anopheles clutzi for paratransgenic approaches.
Good morning, everyone. Today I will present a part of my PhD entitled Effect of Azaya on the Fitness of Anopheles clutzi for Paratransgenic Approaches. It will be presented following this plan. As background, As another presenter said, malaria remains a public health problem worldwide and also in Cameroon in particular. The control of malaria relies mainly on the insecticide-based tools, but these tools are threatened by the emergence of insecticide resistance of mosquito and also the drug resistance. So there is an urgent need for develop novel approach. Nowadays, many novel approach are developed with the purpose to modify, replace or suppress mosquito population. One of these approaches, and not the least, is paratransgenesis, which can be defined as the use of genetically modified symbiont to deliver effectors molecules as and shown at the left to disrupt the transmission of pathogen, and in the case of malaria, the transmission of plasmodium. Previous studies have already shown the effectiveness of this strategy. The case of Serratia, where we clearly show the significant reduction of plasmodium development in Serratia infected mosquitoes. Besides Serratia, another important candidate that has been shown for paratransgenesis is Azaia bacteria. And for being validated as a candidate for paratransgenesis, the bacteria has to be stably associated with mosquitoes, vertically transmission and also transmitted, and also he should not affect the fitness of mosquitoes. Recently, in our study, we have described Azaia bacteria associated with many Anopheles mosquitoes, and we also shown that this bacteria is vertically transmission. But nowadays, few information and no information are available on the fitness of Azaia in Anophel species. That is why we think that after yes, it's okay, thank you. As was saying, after this finding, we think that it's important for us to understand how these bacteria can affect the mosquito fitness and also the plasmodium transmission. But we have started with the impact of the bacteria on life rate of lab rare mosquitoes because of the availability of resources for us. So to answer this question, we use 3 different colonies. One named Azaia minus, where we treated mosquitoes with antibiotic. One where we did not treat it, namely Azaia plus, and one where to take not treated and infect experimentally with an Azaia culture. Prior for the establishment of these colonies, we isolated Azaia to set up our Azaia plus colonies, and also we did an antibiogram to choose the best candidate to inhibit Azaia in mosquitoes. After doing that, we set up our colonies where for Azaia minus, we treated newly emerged mosquitoes with antibiotics for 4 days, and after that, we take a subset of mosquitoes to confirm the efficacy of treatment. For the setup of Azaya plus plus colony, we did it by 2 ways. We infected the water with Azaya culture, and also we fed adult mosquito with sugar supplemented with Azaya culture. After that, as before, we take a subset and confirm by qPCR. When establish our different colony, we evaluate the different life set as such as fecundity, fertility, and longevity. As result, we obtain no inhibition for antibiogram, but we choose to use doxycycline, and we investigate 2 different concentration, 8 and 16. We found that 16 milligram per milliliter were the best, so it's at which concentration that we treated our mosquitoes. And right here, we clearly seen that the establishment of different colonies were successful. When you look at the amount of Azaya in the different group. According to impact of Azaya on fecundity and fertility, we found a difference, significant difference in oviposition rate between Azaya minus and Azaya plus. When you look at the egg laying, we clearly show that there is a difference in the 2 groups, and also the difference between natural Azaya plus and natural plus infected Azaya plus plus. So we think that Azaya can have a positive impact on the ability of mosquito to lay eggs. It's the same case with the hatching rate, where we show Azaya positively correlated to the ability of larvae to hatch. And that is we think this is a good result if you want to implement the population replacement strategy. Concerning the impact of Azaya on longevity, the results shown a significant difference between the 3 groups, with a reduced longevity observed in antibiotic-treated mosquitoes. Once more, we think that Azaya can have a good fitness, and if you want to implement it by population replacement as previously, we think that it can be a good candidate. So after that, we can summarize by saying that this study revealed that Azaya presence or abundance in Anopheles mosquito provides high fitness, confirming that this bacteria is a good pathogen, a good candidate for paratransgenesis. So for next, if you have resources to continue, and we think so, we can have it maybe after this ANTIVEC meeting. We can investigate the impact of Azaya on Plasmodium development and transmission. Saying that, we thank ANTIVEC, we thank our institution, founders and partners. So thank you for your attention.
Thank you, Claudine. That was another lovely talk. Have we got any questions? Just at the back.
Okay.
Or have you got the microphone?
A microphone.
Nice presentation. I have a question.
Thank you. Yes.
Did you check the levels of the bacteria in the water when you're feeding the larvae?
Check in the water, no, but we have the amount of Azaya that we've with infected the water. It's just the running water.
Yeah, that's what I'm asking. Did you check it? Like, is it changing at the concentration? Because that's very important with paratransgenesis application.
Yes, you are right. What we did after culture, we take a culture of Azaya and we infect mosquito at a concentration of 10 exposant. 10 no, not 10%. or the power of 8 colony for unit per milliliters. Yes, that was the concentration. Thank you.
Okay, I think we're going to have to do another question during lunch. Is that okay?
Yeah.
So we're going to go for lunch just now, and if everyone can come back at 1:30 on the dot, please. That's great. Thank you. Thanks.
Yeah. Do you want me to just play the screen too in case? All right. So, testing, testing. Does it come through? So if I were to give a talk like this, you would be able to hear me at the back of the room? Crystal, can you hear me? Does this work for the room?
It works really clearly.
Why don't we just do it that way? Then everyone can go get lunch. We'll just stay here. We'll just, I won't change anything. Can you still see my screen?
We've got it off right now, but we can.
Okay.
Sure.
Yeah, I would say so. Maybe she can speak so that we can determine if the sound is clear, louder enough.
Yeah, we're going to do that just now. Just hold the mic here, try it this way. You want me to speak again so you can test the back of the room? All good?
Okay.
Does it sound okay, Krista? Okay. Yeah. Right. I won't touch anything. I'll just leave it all here, and then when you're ready, I'll see you on here, and I'll turn the camera and sound on and give my talk rapidly as I can.
Okay, that sounds like the best idea. Thank you.
Thank you. Have a good one.
Okay, great. We're going to get started with the next session. We've got a lot of exciting talks to get through, so let's make a start. First up, we have Mara Lonigzak from the Wellcome Sanger Institute, and yeah, talking to us about malaria, Anopheles genomes for malaria vector control. So I'll hand over to you, Mara. We're looking forward to your talk.
Thanks. Jeremy, can you hear me okay? Yeah? Can you hear me? Cool. Shall I get started? Okay. So first of all, thank you so much for letting me dial in. It's been really great to be able to follow all the exciting progress that you all have been doing from the streaming conference on YouTube. I'm going to whiz through this because I gather that there's a couple talks that we didn't hear from yet from the last session. I'm going to talk about two projects in which we really want your participation. So the first one is all about generating reference genomes for Anopheles species. We know that accurate and relevant reference genomes are really key because the short reads that we use to investigate things like heterozygosity, population size, gene drive design, and population structure all depend on having the ability to have that sequence represented in the reference genome. So if it's not there, or if there's a gap, we miss this information. And I also say relevant reference genomes because I think there's a lot of structure that we've discovered in the major vectors that have very widespread ranges, and we want to be able to accurately represent that in reference genomes. Additionally, the last project I'll talk about, ANOSP, has depended on having a bunch of reference genomes in order to design these targeted sequencing approaches. So because of the technology leaps in the last few years, PacBio Hi-Fi in particular, which allows which has a low input protocol, that means that we can create single individual mosquito reference genomes from wild-caught individuals that are higher contiguity and greater quality than anything we've been able to do in the past. So these are about 10 or 15 individual species in which we've generated very high quality reference genomes using single individuals. And I want to point out in particular Mushedi and Nili, which Sandrine Nsengo from Cameroon actually shipped these two species where she punctured them just lightly, squished them lightly in ethanol so that their ethanol penetrated their cuticle. And they came to us at room temperature rather than on dry ice, which saves a huge amount of cost. And the quality of the genomes there is really, really good. So it is now completely feasible to generate reference genomes from very low input. We have a new Gambi reference genome. So this is pest up here in the blue, and the new one called Iphacara strain here in the orange. And you can see these tiny little black dots. There's only 33 of them. And those are the remaining gaps in the reference genome for Gambi, compared to over 6,000 gaps in pest. And additionally, the new Iphacara genome, you can see that it's longer than pest. It has 39 megabases of data of sequence that is now placed on chromosomes. There are no inversions here. These little orange, the structural variation that you can see here may be real inversions, but it may also be just assembly errors in pest. So this is a standard inversion karyotype. We've also recently created a new Funestus reference genome. This is from a wild individual caught in Gabon. There was already quite a good reference genome for Funestus from a Mozambique colony from Fumas. And Fumas is up here on the top, and you can see all these little black dots are remaining gaps that existed in the Fumas genome. And the black dots here, there are very few now in the new wild-caught individual genome. We've marked the four inversions that we're very confident of. So these are already studied inversions with names. And all these other structural variants that you can see here, it's unclear yet whether those are real or whether those are assembly errors in FUMAS. So there's very few gaps, and it's a very nice genome. Please use it. Additionally, in VectorBase, it's now represented. It's got some funny names here, which will be fixed in the upcoming release. So this is the chromosomal name. It's been annotated, the genes are there, and all of the reference genomes that we've created are in this pipeline to get annotated and put up on VectorBase for everybody to use. So what we want from you is that we still need to improve the annotations for some of the species. So if you have samples that you could contribute to this community effort that could be sequenced, RNA sequenced, like for example, Nili and Kustani and Marshale, we really could use some samples that we can generate better quality RNA sequence from. Or if you have RNA-seq projects and process for those that you could allow us to use to annotate these genomes, that would be great. We also have Gates funding to deliver more reference genomes. This is a community, this is for the community. So we really want to know what additional reference genomes you need. We have, for example, the ability to generate more reference genomes from the major vectors, but also for the minor vectors. We're really keen to start to explore and make sure that we have genomic resources for these vectors that are contributing to transmission around the continent, as well as their close non-vector relatives from anywhere. So please get in touch with us. You can use this tiny URL, Anopheles ref genomes, or you can just email me, either way. And if you do provide a sample, you would be the first author on the resulting genome note. So we basically write these little tiny notes that basically reveal the sort of process of getting the genome into place and the quality of it, and a little paragraph on the species. So the major project that we're undergoing now is called ANOSP. And ANOSP is like a multifunction tool, similar to a Swiss Army knife. So this is our little logo. Go and see Amjad there at the conference if you want a sticker. It is a high throughput multifunction surveillance tool that we are using to study species diversity and transmission potential at very large scale. So we have funding and capacity to run 500,000 mosquitoes through this process. We really want to partner with people who would benefit from it. It is applicable to any Anopheles samples, so while our focus is on adults, it could work on larvae, on bulk traps, or on environmental DNA from larval breeding sites, for example, if you really need to know what species is breeding in particular pools. We use the Lysis-C SOP, which is available at protocols.io, where we put individual mosquitoes into plates. We non-destructively extract them so their carcass remains for further morphological examination. It's very cheap. The main cost there is around the plastics and the proteinase K. We use just 0.1% of the DNA that we extract, so all the DNA is basically there still for additional sequencing. Whole genome sequencing works on Lysis-C extracted mosquitoes. It's very high throughput. We're doing 800 individual mosquitoes per MySeq run. And the reason that this is beneficial is because it creates a common language. You don't need to know anything about it other than it's an anopheline to put it in there. So there's no sort of bespoke, the Phonestis complex PCR protocol, or the Phonestis or the bespoke PCR protocols to try to figure out what is the species that you're looking at. You don't need to do that anymore. This will work if you want. Additionally, it's got 2 markers in it, so I'll switch to this. Alex and Petra in my group were really key in designing this a couple years ago. It's got 62 kind of generic loci across the Anopheles genome. And in total, it's just 10 kb of data per individual. These are each 160 bases worth of targets. It's got 2 additional loci that target the generic Plasmodium mitochondria, so it tells us if that mosquito is carrying parasites, and if so, what species of Plasmodium it's carrying. It's 1 PCR per individual, so it's a PCR reaction that does 64 primer pairs in one go. And this is kind of you can read this paper if you want to find out more details, but this is kind of the recovery we get. So even when we get very genetically distant from Gambi complex down into the Curtesia subgroup, we still are getting 30 amplicons there, which is plenty to tell the species. This is an example of what the Lysis-C protocol looks like. So these are individual mosquitoes in the well. And I won't belabor this. You can look at this protocol here. It works out to be very cheap, and you keep your specimens, which is really nice, much cheaper than kits. Key to our approach here is that we standardize metadata. So we are very happy to accept specimens from any project. We require at least 800 specimens to get started because that's what goes into a run. And we require each individual to have standardized metadata here about when you caught it, where you caught it, what piece of the organism you're giving us, whether it's just the head or the whole body, etc. So I'm going to go quick through this because I know we're behind time. This is now published from Marie-Lou Baudet. We've created a sort of analytical approach that we call NOVI. The NN stands for nearest neighbors, and that's the first step here. So the protocol works because it doesn't use alignments. It uses k-mers. So it breaks each of those 62 loci into 8-mer pieces and doesn't require alignments between very distant related species. And instead, it requires the k-mers that we identify in any one individual to match the reference database that we've created from well-curated individuals. So the darker colors here mean higher k-mer similarity. And you can see this big square here is all Gambi complex group. And many of these other small kind of triangles along here are all 62 different species from across Anopheles. So we're keen to get every species represented in the database. If you're working with non-vectors or vectors that you know what they are, please get in touch because we would like to have good representation of everything. So the problem with the nearest neighbor approach, and the reason it's called NOVI, because the second stage is a VAE for the Gambi complex. And that's because these are too similar to each other, Gambi and Kalutsi, to identify with a nearest neighbors approach. So we use a variational autoencoder, which is represented here in a low dimensionality projection. So this is basically our reference index for the Anopheles Gambi complex. And we did not tell the machine learning algorithm what species was what. It just took all the data and it created this structure. And then when we layered on the species names, you can see that we're able to differentiate Gambi here in blue from Kalutsi here in orange, as well as all the other species present. So what we do now is when we have a new species, a new individual that comes through, we identify it using the nearest neighbor approach as being Gambi complex, and then we have to project it onto this VAE to say which individual species is it within the complex. This projection also hides some of the geographic structure that we know to exist. So you can see some of the continental structure here represented in the colors here for Gambi, where you have Madagascar here and south and east here, and more west and central here. Additionally, as I mentioned, there's 2 plasmodium mitochondrial markers in the primer mix. We have also a reference database of all haplotypes for plasmodium, and we are working on refining this approach. So here's an example of a real data set sent to us by a member of somebody in the audience there, Mahamadi from Target Malaria, who sent us 10 plates, so nearly 1,000 individuals. These had actually been stored for quite a while in groups in ethanol. So he plated them all out for us. We buffer C extracted them, and you can see the projection here. So these are all individuals that turned out to be Kalutsi, Gambi, and Arabiensis. Two of the 44 Arabiensis, you can see the numbers here. Some of these showed potential parasite presence, and Mahamadi included information around whether there was a blood meal present or absent, and you can see a much higher proportion of individuals that actually have plasmodium detection in individuals among those that have blood meals, which means that they've probably just recently found out a positive person rather than actually necessarily contributing to transmission. We also saw 3 different species of plasmodium among these samples for human plasmodium, as well as 2 zoonotic species, I think a goat malaria and a buffalo malaria. This high quality metadata that's well curated allows all sorts of different studies. So this was outdoor collected mosquitoes from Mahamadi's project. They were recorded through the night and then put into these epidorfs in groups of 10. So he knew exactly which hour each one had been collected, but not which species they were. And you can start to see different patterns about which species are coming in through the night to seek hosts. So we want to work with you. We need more species to improve our reference index, especially where the species ID is confident. So you know it's the sample, you know it's the species. We really would like to get 10 individuals of every described Anopheles species for our reference index. Additionally, if you need species IDs on thousands of Anophelines and you're willing to put them into plates, because we can't accept epidorfs, collect that careful metadata and complete the ABS procedures with us, we can generate ANOSP data for your project. We can also generate CL1 if you need it, and we return all data together to you, together with a report that looks like this. So you can't really see the details here, but this says, this is what was submitted by Mahamadi, and these are all the different species. So although most of them were thought to be Gambi complex, we did detect a couple of nili there and some other ones that still require further work to get a name. So again, there's a tiny URL, ANOSP, where you can register your interest, or you can just email me. Our longer term plans are that as I said, we need to grow the reference database. We're working on a version 2 that will incorporate Cas9 detection for the gene drive efforts, which means that as gene drive release happens in the future, we can monitor not only its spread within species, but its spread between species. We're working very hard now to simplify and make the full wet lab and analytical workflows accessible to anyone. We cannot run this assay in my lab. It is not built for a standard molecular lab. It's run on high throughput robots right now. So we're working to reverse engineer it to make it work in a standard lab, and we hope to have that completed within about a year. We want to create a very user-friendly, completely open access data analysis ecosystem that encourages data sharing, because of course mosquitoes don't respect our geographic boundaries. And we're interested in creating additional partnerships, so get in touch with us. Thank you to these folks from my group who were really working very hard on creating this whole process, and these folks here who have already contributed samples for ANOSP. And I'll end there. I also just want to draw attention to this thing at the bottom here, which is a collaboration between PAMCA and MalariaGen, working on really an incredible training set of training modules where you can actually go and watch videos and learn how to do genomic analysis on the data that's out there. So highly encourage people who are just getting started and looking to improve their analysis skills to check that out. Thank you very much.
Great, thank you very much, Mara. That's a real great resource for the community. Do we have any questions for Mara? Yeah, one at the back there.
Hi, thank you so much for great work and for new reference genomes. We were really needing them. So I just wanted to ask regarding Y chromosome. Have you maybe saw any longer contigs than they were previously in the Y unplaced in pest genome? And how, in what state the Y chromosome is in the new reference genome?
So I think the question, it's hard to hear a little bit. I think the question is about why Y chromosome contigs in the reference genomes, and we are seeing them. We intentionally tried to sequence so for many of the samples we were provided in the first round of this project, it was a wild-caught mother and her brood. So we had male and female siblings that we actually put through the whole process, and we are getting Y contigs. I can't remember off the top of my head how big the biggest one is, but we have high confidence that they're Y because we have the brother and sister sequenced. And that's a kind of continuing effort. If you want to get in touch with me, I can tell you more details. I just don't have them to fingertips.
Great, thanks. Any other questions? Maybe time for one or so more. I just maybe very quickly, the sample shipment on dry ice and stuff is really tricky. So that's where it was really nice to see that you got such a good genome from a room temperature. Can you tell us a bit more about how that was actually achieved?
Sure. So yeah, I skipped over a lot of details, but actually when we first were working with Sandrine, it was going to cost £4,000 to get the dry ice shipment from Cameroon to Senghor, which is more than the cost of the genome. And we had been trialing something in the lab where we actually, so we take a mosquito and we put it live into ethanol, or we kill it basically in ethanol, and then we puncture it. You can either do this by taking a blue pestle, the ones that you typically grind the mosquito with, and kind of gently squishing the mosquito against the wall of the Eppendorf in ethanol. You don't want to squish it too much because you don't want all the cells to come out, which means all the DNA leaks. You just want to compromise the cuticle. We've also had good success by taking a syringe and just puncturing the thorax once so ethanol can penetrate. Shipping at room temperature without doing that does not result in a good high molecular weight DNA. So that is really key. Ethanol, we use 100% ethanol. It can also, I think other preservatives are working well too, like many of the ones that actually cost quite a lot of money that you can buy. I can't remember, DNA shields, etc. Those all work. But the puncture is the really important thing, the puncture or the squish.
Great,
so
yeah.
if you are, I guess I should say, if you are out and you're collecting and you're thinking of contributing to this, for the reference genome, that's absolutely key. We can't take kind of, if they haven't been snap frozen or they haven't been punctured in ethanol, the DNA quality won't be good enough to create a reference genome. For ANOSP, it works on almost, like you barely need any DNA there. It works on a leg, it works on a really old stored mosquito. It's much more sensitive because there's a lot of amplification that happens through the PCR process.
Great, thanks, Mara. I think we'll move on then to the next talk. Thank you very much for that. It's a great talk. Okay, next speaker is Juan Masquerda, who's going to be talking to us about transmission blocking in ticks. So it's great to have a bit of diversity with the subject here.
Hello, hello.
Yeah. Good afternoon, everybody. I'm going to change gears a little bit here, talking about our research in my laboratory at the Autonomous University of Querétaro, about our efforts to develop transmission blocking strategies of babesiosis in the tick vector. I'm going to start by saying that 80% of the cattle in the world is exposed to ticks, and that ticks not only cause damage by feeding on the blood, but by injecting toxins and by especially transmitting pathogens. And among these ticks, one of the most important ticks in cattle is the Rhipicephalus microplus, known as the cattle fever tick, because it transmits the cattle fever disease caused by bovine babesiosis. It can transmit Babesia bigemina, Babesia bovis, and Anaplasma marginale, at least in the American continent. In some other continents, it transmits some other species of Babesia. This is a female, 25 days post-infestation, when it acquires more than 1 milliliter of blood. And this is the male when it mates. And then the female detaches, ovopocytes 3,000 eggs. The eggs become infected by transovarial transmission, and then the female dies, and the larvae are infected. We've been observed that also the tick is affected by the babesia, because when the tick feeds on a high amount of infected blood, then the tick may die, meaning that there is a very close interaction between the parasite and the vector. We have tried to develop vaccines against bovine babesiosis. We have already developed a vaccine basically focused on the asexual reproduction, but the sexual reproduction, which takes part on the tick, is the subject of the research that I'm presenting right now. Long time ago, we developed a culture where we were able to induce sexual stages of babesia out of the without tick content. These stages that we call sexual stages, we developed antibodies against them, and these antibodies could recognize the stages in the midgut of the ticks, meaning that they could share the same antigens. Also, we characterize a gene that is the HAP2 gene. It is a male gamete gene required for cell fusion. Our group and some other groups show that this gene is not expressed on the asexual stages, but on the sexual stages. And we show that antibodies against HAP2 can block zygote formation in vitro, meaning that it could be used as a vaccine target. So we try to focus on transmission blocking vaccines, basically to prevent vector infectivity and to reduce transmission. As you may know, there are several proteins that have been characterized in the tick pathogen interface that some other groups have been working, but we focus on interactomics. What we did some time ago is that we labeled live babesia. We exposed them to proteins of the midgut tick by an overlay, and then by mass spectrometry, we identified those proteins of the midgut of the tick that interacted with babesia. We identified especially one protein that we call a voltage-dependent anion selective channel that is called VDACT. And we saw that VDACT is a important protein that is upregulated when some parasites are infecting their vector or their host. So we wanted to know whether there was an effect of Babesia bigemina on the expression of this protein in the midgut of infected ticks. We showed eventually that infected ticks at several times after infection could upregulate the transcription of this gene, not only the transcription, but also the expression by Western blot analysis. And also by confocal microscopy, we show that this VDAC protein not only increased expression, but also translocated to the membrane of the infected ticks, of the infected cells of the ticks. So was this protein a vaccine candidate? For cattle, we expressed the protein as a recombinant protein with a histac, was purified, and then we used in a very complex experiment. I won't have time to explain it, but we had 3 groups of 5 cattle. We immunized some of them with the protein, some others were not immunized. They were infested with ticks, and some of them were infected with Babesia. And then we obtained the efficacy of this vaccine, which we use a formula that is already characterized. When we observed that the cattle that were immunized with the protein but were not infected with Babesia, the vaccine had an efficacy of 88%, which is higher than all the other vaccines that have been published previously, even the commercially available vaccine based on BM86. But the cattle that was immunized but infected with Babesia, the efficacy was low. Because of that, we included HAP2 in a multi-epitopic chimera vaccine that we tested already and that we showed that had a good efficacy. We had applied for patent several years ago. And I just want to find out, to finish by saying that recently we are working in a collaboration with researchers from France, trying to develop or to characterize microbiota in the tick that enhances Babesia transmission to develop anti-microbiota vaccines. And with that, I appreciate your time. Thank you.
All right, thank you very much. Do we have any questions for Juan? Yep.
Thanks for that. Just on the vaccine, the trial you did there with the vaccine, I didn't understand the difference between, you said infected, and there were 2 categories, 23% and 88% protection.
Yeah, one group was immunized with the vaccine and challenged with the ticks.
With tick, and the other one was injected.
The other one was challenged with the tick, but infected with Babesia.
Challenged with the tick. So how was? Okay, maybe this is shut off.
Yeah, I know it's complicated because the vaccine works against the tick.
Okay, right.
But the cattle are infected in the field, so we have to also get it infected with Babesia.
Okay. Any other questions? Good. Okay. Great. Thank you very much, Juan. great. Next we have Shun Olivier, who will be telling us about pollution and how that's affecting malaria vectors. So over to you, Shun.
Good afternoon, everybody. My name is Shanae Oliver. I am from the National Institute of Communicable Diseases and Wits University, and I am finally closing off the session that was meant to have finished at lunchtime. Okay. So I'm fairly certain that I don't need to introduce the concept of paratransgenesis after all the talks that we have done this morning. But what I would like to bring your attention to is that when you are choosing your relevant symbionts, you can either make a move to use genetically modified symbiotic bacteria, or you can use naturally occurring symbiotic bacteria. Now, because I am at the beginning of my project, and what I'm essentially doing is cataloging the bacteria from my native Anopheles arabiensis, you can guess which one I'm going for. Each one has its pros and cons, but because I'm looking at native symbiotic bacteria, I am interested in what the environment that these mosquitoes find them in is going to do to the gut bacteria. And the reason why I'm interested in this is because my group has already shown that larval salt exposure can alter the gut microbiota of Anopheles murus. So as we have heard earlier on today, Anopheles murus is an osmotolerant minor vector, and what we have demonstrated by 16S sequencing is that larval exposure to varying salt concentrations altered the diversity as well as the relative abundance of species that are relevant to paratransgenesis in the adult mosquito. Now, the reason that I am concerned about this is because of previous work that I have done. Although in general, members of the Anopheles gambiae complex breeds in clean, clear, sunlit bodies of water, this is changing, and they are adapting to breeding in polluted water, and this is particularly true for Anopheles gambiae and Anopheles arabiensis. So before I started working on microbiomes, I was working on pollution, and we had shown that larval pollutant exposure had a major effect on the life history of the adult mosquito. And because most of my work focused on the effect of heavy metals, I decided to look at what heavy metals does to the gut microbiome. And what I have basically done is that I've looked at the effect of cadmium and copper on 3 strains of mosquitoes, an insecticide-susceptible strain of Anopheles arabiensis known as SEN, an insecticide-resistant strain called SEN-DDT that was selected from the SEN strain, and finally, F1 mosquitoes from a local population. Now, I know that not everybody is familiar with microbiomes and how they work, so I just wanted to give you a little bit of a primer on analyzing the microbiome. When I talk about alpha diversity, I'm talking about the diversity within the sample. When I talk about beta diversity, I'm talking about diversity between samples and treatments. When I talk about overlapping species, I'm talking about bacterial species that are shared between the strains. And finally, when I talk about differential abundance, I'm looking at which bacterial species are more abundant in each strain. So when it comes to alpha diversity, there are 2 ways that you can look at the diversity of a sample. You can either look at the sheer numbers of bacterial species that you have, and this is known as species richness. Species richness can be measured by the indicators ACE and CHOW1. And you can also look at the way the bacterial species are distributed, and this is known as species evenness. And species evenness is measured by Shannon and Simpson indices, at least for what I'm doing. So if we take a look over here at the metal effects on alpha diversity, what you may notice, if you could see it clearly, is that there's actually no difference in the alpha diversity of the insecticide-resistant and the insecticide-susceptible strains, here in red and green. But when you compare those to the F1s, we see that the F1 alpha diversity is affected by the toxicity of the metal. So basically, the more toxic the metal, the greater the effect we see on the F1 strain. Now, when we look at beta diversity, we see that there is a vast difference in the beta diversity of the laboratory strains, which are these tiny little variants over here, compared to the F1s. And as you can see, the F1s have a far greater beta diversity. What you should also notice is, although there is overlap in the untreated, which is the big red triangle over there, and the two different treatments, although there is overlap, we can still see that the metal treatment has a significant effect on the beta diversity. So when we look at the overlapping species, we see that the untreated adults have by far the most unique species. This is followed by copper, which was less toxic than cadmium, and cadmium, which was quite toxic to them, had the least number of unique species. And when we striate this out into the different strains, we see exactly the same pattern, with the untreated having the most, copper having the second most, cadmium having the least, and in some cases, no unique species whatsoever. Now, I know this is quite a busy graph, but all I want you to take a look at is this is differential abundance when we between the laboratory strains and the F1 strains after treatment. So generally, on the left, we will have the laboratory strain, and on the right, we will have the F1 strain. And what you will notice here is that regardless of whether you treat it with copper or cadmium, when compared to the insecticide susceptible strain, the F1 has more species that are differentially abundant. However, this pattern changes when you compare it to an insecticide resistant strain. Here, it flips around, and the F1s have far less species that are abundant. So what this suggests is that the greater the resistant phenotype, the greater the number of bacterial species that will be abundant after larval metal exposure. Also, although you cannot see, it's not just the number of species that are relevant, it is which bacterial species that are abundant after treatment is very strongly affected by the insecticide resistant phenotype. When we look at the F1 alone, again we see that the highly toxic cadmium treatment results in less species being differentially abundant compared to the less toxic copper treatment. So in summary, larval exposure to metal pollutants has a very marked effect on beta diversity and the relative abundance of bacteria. What is very clear is that the insecticide-resistant strain had far more bacterial species abundant after larval metal exposure, and this may simply be because the insecticide-resistant strain is in general more tolerant to toxic pollutant exposure, and this may underlie this effect. So, for those of us who are still looking and cataloging bacteria, the abundance of bacterial species with known paratransgenesis potential was affected by both insecticide resistance phenotype as well as the larval metal treatment. So if you are maining for native symbionts for paratransgenesis potential, the local larval environment must be considered. I would like to thank my funders for the funding. Although sequencing is becoming cheaper, it's still not for free. And of course, the people who have done the work, my students and the people who are involved with the core sequencing unit. And finally, for the F1s, we would like to thank Dr. Giv Munenga and the SIT team for the collection of the material. And finally, if I can just do one last point, if you're working on symbionts, and clearly there's a lot of people, if you are considering somewhere to submit your work, please consider submitting it to the special edition in the MDPI Journal Tropical Medicine and Infectious Diseases. If you are interested, please give me a contact. Thank you very much.
Thank you very much, Shune. That was great. Any questions? I might quickly ask one then. So do you think this effect is through the mosquito, or is a direct effect of the on the bacteria, as in the metals in the water affecting the bacteria growth in the water?
This is something that we are currently investigating because firstly, we are comparing it to different water treatments, and we are trying to look at how it affects bacterial dynamics. But in all honesty, I think it is both. I believe that the actual treatment of metal alters the bacterial population in the water, which is something that we are still investigating. And I do suspect that it's also going to affect the dynamics because along with the larval metal treatment, we have upregulation of detoxification genes, immune responses, and all these things are going to have an effect on the bacterial dynamics. So I think it's a combination of both the environment directly itself and what the environment is doing to the mosquito's physiology.
Yeah, great. Any other questions? much.
Session closed.
Thank you. All right, great. So I think it's our first Wolbachia talk, I'm not sure, but we'd like to welcome Tom Walker, who's going to be telling us about Wolbachia strains in Anopheles mosquitoes. So looking forward to that. Thanks, Tom.
Thank you, Jeremy. Good afternoon. So I just want to introduce myself in terms of where I'm at. I'm at the University of Warwick now. Previously, I was at the London School of Hygiene and Tropical Medicine for 8 years with a Wellcome Fellowship. And before that, I was in Australia with Scott O'Neill doing a postdoc. And then a long time ago, I did my PhD with Steve. So, sorry, Steve, that probably ages you a bit, doesn't it? I couldn't get away with talking here with just about Anopheles. So I've started with some of the work that was done in Australia with Aedes and dengue virus, because it kind of sets up the rationale for why we want to do this work with Anopheles. And I went to Australia with the idea of trying to infect Aedes aegypti with different strains of Wolbachia. And the one we settled on was a WML strain, which is in Drosophila fruit flies, so it's found in lots of different places. And we used micro-injection of embryos to create these infected lines. And what we see when we get these stable infected lines is co-infection where we put in 2 Wolbachia strains. So initially I created a single infection, the WML line, and then I put a second Wolbachia strain on top to create a double infection. And what we find with both the single infection and the double infection is they're maternally transmitted at close to 100%. And we obviously needed to look at whether they could spread through mosquito populations, because Wolbachia, if you're going to use it for biocontrol, you need a mechanism for it to invade populations. So this is something that I hope most people are familiar with. It's cytoplasmic incompatibility. And that's where the female has disappeared on the slide, but she is there. She's uninfected. And when she's mated to a male in blue, you get no progeny. And the resulting crossing types results in a reproductive advantage to have Wolbachia. So we found that the WML strain induced CI on its own, and the double infection induced bidirectional cytoplasmic incompatibility. And what this would mean is the double infection would spread over the top of the single infection in the field if we needed to release a second strain. Another very important thing to look at when you're creating Wolbachia infected lines is what impact does it have on host fitness. So all the measures that we did showed really the single infected line and the double infected line had minimal impacts on host fitness. So we predicted that in the field, if we needed, a superinfection would replace a single field infection. So that's great. The Wolbachia strains can invade mosquito populations, or they're predicted to, but can they inhibit pathogens? So the first thing we did was we fed mosquitoes that had Wolbachia and didn't have Wolbachia with dengue, infected blood, and we saw by just simple quantitative PCR that there was a big effect on the amount of virus present in both whole body mosquitoes and in legs, which were a proxy for dissemination. But we wanted to go one step further and show that Wolbachia can inhibit dengue virus in saliva or in the salivary glands. So we did some forced salivation work where we got mosquitoes to spit into tubes essentially, then collected their saliva, and then used immunoplaque assays to look for the presence of infectious virus. And what we found was 100% of the mosquitoes that had Wolbachia blocked dengue transmission. In other words, there was no infectious virus coming from these mosquitoes. And then some work done a little bit later on the superinfected line, where we fed mosquitoes that had Wolbachia and not Wolbachia onto patients that had dengue in hospitals in Vietnam. And we were able to demonstrate that there was a greater reduction in the percent of mosquitoes that had dengue in the saliva with the super-infected line. So it seems like there's an accumulative effect of 2 Wolbachia strains are even stronger than a single infection. But this is really where the World Mosquito Program is now. So it's turned from the work that I did in the lab and others did in the lab, obviously, into a big project where the WML strain has now been released into 11 different dengue endemic countries. So the World Mosquito Program is now looking at whether it inhibits dengue virus in field populations. And you can see on the top right there that the vast majority of locations where it has been released, you can't really read the locations, unfortunately. I picked the wrong color, but it's very high apart from in certain places, which 40% is Rio de Janeiro. But the proof was really published 2 years ago now from a cluster randomized trial. So this was undertaken in Jakarta City in Indonesia, and that's where they had 12 intervention clusters, so the clusters that had the Wolbachia infected mosquitoes, and 12 control clusters with no release mosquitoes. And the effect was quite dramatic on the number of clinically confirmed dengue cases. So they had a nearly 80% reduction in the incidence of virologically confirmed dengue, and about 80% or even higher of hospitalizations for dengue virus. So it had a big effect on the amount of dengue being transmitted. And I moved back from Australia in 2014 to start my lab at the London School of Hygiene and Tropical Medicine. And around that time, there were a few papers being published on whether Wolbachia is in Anopheles. So there are a few studies using standard PCR or even nested PCR to show that there's probably evidence of Wolbachia in wild Anopheles populations, which we didn't historically think was there. So had technology increased so we could detect these strains, or is there something else going on? So we set out to collect mosquitoes from 5 different malaria endemic countries. So I can see my screen here, but you can't, so I don't know why I'm continuing to talk. Okay, I'll give it a go and see how we do. So I was saying that we were collecting mosquitoes to see if there was further evidence for the detection of Wolbachia in natural populations. So we collected different mosquitoes and actually got some collections from collaborators. This wasn't all new field work. But what we were able to do is demonstrate the presence of possible strains in a few different mosquito species. Anopheles mouchetti was the first one that was very obvious. And then we did find it in Anopheles gambier and arabiensis, but our PCR data was a little bit patchy, which I'll talk about later. We also found in another species that our best attempts were on sequencing was Anopheles species A, so it was really unknown at that point. We went back and did further field work in Guinea, and we were able to detect Wolbachia in Anopheles melas, in Anopheles melas gambi hybrids, and in a single individual Anopheles species that we still don't know what it is, because it's only 93% sequence identity to the nearest GenBank accession number. So we're still really not sure what that species is. And I've included these quite complicated phylogenetic trees because this was our way to really demonstrate whether we thought we had a genuine infection. So what this is sequencing of all 5 MLST genes, which is used to type Wolbachia, and you create a super sequence, and then you put them in phylogeny. And this allowed us to really determine the phylogeny of these different strains. And then you can look at the database for Wolbachia and really determine what kind of strain you're looking at. On the left-hand side here, it's pretty hard to see, but for Anopheles species A and Anopheles mouchetti, so that's red and green, they generated novel MLST allele numbers. Anopheles kulutzi matched pre-existing allele numbers, but the combination of the 5 was novel. So we were a little bit unsure as to what was going on there. The same scenario essentially is happening on the right-hand side. Anopheles melas produced 5 matches to pre-existing alleles, but just in a combination that would be considered novel, whereas Anopheles species X, the unknown mosquito species, produced completely new novel allele numbers, so that we put into the database. So with this preliminary data, we decided to focus on 2 Wolbachia strains in 2 different mosquito species. So we wanted to provide further evidence that these strains were in genuine endosymbiosis with Anopheles species. So we focused on the 2 Wolbachia strains that our PCR data indicated would be more likely to be genuine strains in endosymbiosis. So we focused on Anopheles mouchetti from both Cameroon and the DRC, and also Anopheles demolini from the DRC, and also historical samples from Kenya. On the right-hand side, we did some ITS2 sequencing to really confirm our ideas on both morphology and species ID using standard PCRs. And what we went on to do is try to prove, as best we could, that these strains were in genuine endosymbiosis. So we did some quantitative PCR. To our knowledge, no one has been able to pick up high levels of Wolbachia using quantitative PCR, and we found quite high levels in both Kenyan and DRC populations. And we also chopped off the head thorax for Anopheles mouchetti coming from Cameroon. That gave us quite high levels of detection in the head thorax, which is evidence of a somatic infection. And then we did some fluorescent in situ hybridization to prove that there is live bacteria in the ovaries of Anopheles mouchetti. We also sequenced the genomes. So we took the whole mosquitoes and we put them through Illumina sequencing. And I'll talk a little bit about this later, but this table shows you the difference between Anopheles mouchetti and Anopheles demolini versus in the Anopheles gambi complex. The reads that we generated from mouchetti and demolini sit within known Wolbachia infections, whereas those from Anopheles gambi from Burkina Faso and Anopheles kulutzi that we collected and Anopheles gambi we collected from the DRC. So they're mapping that the amount of Wolbachia reads is much, much smaller than when you compare it to the mosquito genome reads. And this is backed up by some microbiome analysis. So we took some mosquitoes and did some microbiome analysis, and this is sort of raw data we would get on the left-hand side with Wolbachia being pink. And that's translated up to the top right-hand corner here. That's the percentage of Wolbachia reads versus in the whole genome, sorry, in the whole bacterial sequencing. So here you've got a clear case where Wolbachia is present, it's the dominant bacterial species. This is the case in both Anopheles demolini and also in Anopheles mouchetti. And I've just included this other result that came from a paper published around the same time. This is on Anopheles kulutsi, where the title was the microbiome and its interaction with the intracellular parasite Wolbachia. They found one mosquito with Wolbachiarids, and it was 0.04% relative abundance. So a big, big difference between what we were finding. So we attempted to colonize both these mosquito species in the lab, and we found evidence of maternal transmission. So we got 80 to 90% prevalence in the F1, F2s for both Wolbachia strains. We found quite low densities in the F1 larvae and pupae, but this could simply be threshold detection levels for PCR. So we're currently trying multiple experiments to colonize both strains within the two mosquito species. And we also did a little bit of phylogenetic comparison between different geographical populations. The theory being that if there is Wolbachia there and it's an endosymbiont that's been in evolutionary association for a long time, you should be able to detect it in vastly populations that are a long way apart. So for example, we looked at the WSP gene between Cameroon and the DRC, and we found the same 3 specimens from each location with the same 3 SNPs. The same sort of scenario we found with COX-A, which is one of the MLST genes. They have similar SNPs in the similar percentage. So clearly the same Wolbachia strain is being present in vastly geographically dispersed populations. And this kind of just summarizes the genomes, breadth and depth coverage of the genomes. So when you map it against the mosquito genome, you basically don't see anything in the Anopheles gambiae complex, whereas Anopheles demolini and Anopheles mouchetti are the 2 that sit with other known Wolbachia infections in mosquitoes, such as Aedes albopictus and Culex kincaid fasciatus. And this was done by a bioinformatics postdoc at LSHTM. And this is taking the entire genomes and mapping them to look at the phylogeny. It's called FAST-ANI. And what was interesting with this is that the 2 Wolbachia strains in Anopheles were confirmed to be Wolbachia supergroup B, but they also looked incredibly alike in terms of their similarity versus other Wolbachia genomes. So we tried to investigate this a little bit further because this was a quite interesting result. So we did some comparative genomics looking at the Wolbachia strains. It turns out that they both sit within a clade that includes some Drosophila Wolbachia strains and Drosophila similans, but they're not next to each other in the terms of similarity. There are other Wolbachia strains that sit between them within this clade. A couple of interesting things about the genomes. They're incredibly small for insect infecting Wolbachia strains, and they don't really have prophage regions, which was quite surprising. It turns out that the Drosophila Wolbachia strains within that clade also don't have prophage regions. And then Shannon, who was the postdoc, went on to look at the metabolic pathways, and the orange is the 2 Wolbachia strains of interest, and you can see that they're basically maintaining similar metabolic pathways. So it's the genuine Wolbachia strains as far as we can tell. Now, when we looked at the genetics of these CIF genes, so CIF genes are found in all Wolbachia strains that induce cytoplasmic incompatibility. So we could use the genomes that we'd established and look for CIF genes. As I've previously mentioned, there were no prophage regions, but the CIF genes were there, but they're not perfect in terms of evidence that it's inducing CI. So, for example, the strain in Anopheles demolini has encodes a multiple stop code on a frame shift interruptions. So we've got evidence that they're there, but does this actually show that they're inducing CI? And really, that's part of what we're doing at the moment. Is we're doing further collections. We're now working with Charles Wanji in Crid in Cameroon and Marcel to try and see if we can establish colonies in Cameroon. And we're working with Janvier in the DRC to see if we can establish Demolini. And this involves both trying to colonize the mosquitoes, but also to try and establish cell lines. This might be a way to obtain the Wolbachia strain for further work. And then downstream, we want to look at all the characterization work that we need to do. And this is kind of just the progress that we've done. So we've concentrated at the moment on the W and D strain in Demolini, and Janvier, who's sat in the audience somewhere right at the back, I can see him. He collected mosquitoes from Luiro in the DRC, and we found a way to get them to the UK through personal transport, which is much more effective than shipping, it turns out. Obvious, but getting from the DRC to the UK is not particularly easy. So Janvier was able to collect them. Then we used a cell line technique. We used a live dead stain to show that there's still a live Wolbachia in these eggs. And Mukund, who is a postdoc in Grant Hughes's lab at Liverpool, has been able to demonstrate by PCR that we've got infection at passage 13 to 14. The top right is not particularly clear, but we've got Wolbachia there, but it looks like the cells are lifting from the monolayer. And apparently, this is quite common when you have high infection rates. So we're trying to kind of stabilize the infection and get more consistent monolayers. And I just want to talk for the last couple of minutes on some ANTIVEC funded work. So Janvier Bandubone, who is at the back there, has got a poster here. And this was funded by ANTIVEC, and we were looking at whether Microsporidia or Wolbachia could be detected in Anopheles gambiae from the DRC. We didn't find any Microsporidia, sorry Jeremy, but we did find a little bit of Wolbachia. But one pool was super interesting. It amplified WSP, and it amplified 3 of the 5 MLST genes. All the Sanger sequencing came back to W and D. So my first reaction was, it's Demolini. So what did we find out? ITS2, and obviously Janvier's brilliant morphology said it was Anopheles gambiae. ITS2 says it's Anopheles gambiae. Sign 200 says it's Anopheles gambiae. Everything says it's Anopheles gambiae. So what is happening here? If we had a single Demolini in a pool, because remember these are pools of 3 mosquitoes, all my experience with ITS2 would tell me you'd see a mixed sequence, and you wouldn't just get 100% gambiae. So it turns out that Anopheles demolini has been collected alongside Anopheles gambiae. So are we getting some environmental DNA contamination? And could this possibly explain why sometimes we detect Wolbachia in Anopheles gambiae? And that's it. I'd just like to acknowledge staff previously at London School, a whole load of graduate students that collected mosquitoes. Emma's sat here, Bethany is somewhere, and Cher, who's also in the audience, who is going to be working with me, hopefully on Demolini in Kenya. And all the funding bodies, particularly the Wellcome Trust, who've funded me for the last 8 years. Thank you.
Great, thanks a lot for that, Tom. Questions? Okay, we've got one there and one over here.
Thanks very much, Tom. That's great. So was there just one pair of cysteines, and did they have functional domains like nuclease or deubiquitinate?
There is the first Wolbachia strain in Demolini has 2 sets, but in Muschetti there's only 1 set. But neither have what we considered functional set. In other words, there are stop codon interruptions. So,
which are upstream of what would be the functional domain.
Yeah.
Okay.
So we're doing further work on that, but the long story short is we need to colonize and do crossing experiments because it's not completely obvious that they're CI inducing based on these genes. And the fact they're not in prophage regions as well is a little bit strange as well. So that's kind of what we're looking at from the genomics point of view. But ultimately, obviously, colonization would tell us if they're actually inducing CI. Yeah.
Hi, great talk. I wonder if you were looking also in other species within the Muschietti complex.
Yeah, we only found, so we did the PCR, and it was pretty much only Muschietti that we got from Cameroon. Marshalli we got, which is kind of close related. We got a lot of Marshalli, and they were all negative. Negative in the sense of we consider it negative when there is if it produces 16 S band, we call that negative, because if it doesn't go on and demonstrate all of the other genes. So as far as we can tell, it's in Muschietti, but it could be in more closely related species as well, for sure. But it's Muschietti SS, the main Muschietti, yeah.
What do you think is causing the cells to suspend? Is it the Wolbachia or another infection?
Yeah, it's a good question. So Grant Hughes, who I work with as a postdoc in the States, had this same thing where when the Wolbachia is heavily infecting the cells, it causes them to rise up from the monolayer. So we're not 100% sure if it's just that, but the idea is that we're transferring it to different cell lines to see if that effect would consider. The biggest issue from the very beginning is contamination. So you have to use eggs. You can't use larvae for cell line work because they're contaminated. So the problem is getting fresh eggs that are surface sterilized from locations like the DRC to the cell culture labs. So it's quite a difficult process, but we think it's Wolbachia orientated, this rising up of the cells. They were Anopheles gambiae cell lines. So is it SWA B? Yeah. So we've got a couple more Anopheles gambiae cell lines that we're going to try, and ultimately we might try and put it into an Aedes cell line. It might be, but the theory is if we've got it in an Anopheles cell line, it'll be easy to trans-infect Anopheles gambiae, as I did with Aedes cell lines in Australia.
How do you sterilize? I mean, what do you use?
100% ethanol, bleaching, we're worried that it would have a bigger detrimental effect on the Wolbachia. We're also trying things like not cryopreservation, but cold storage temperature to delay the hatching, because often when we send them on from field locations as eggs, they hatch on transit. And if you've got larvae that are hatched, then contamination is the big issue for the cell line work.
Great. Okay, thanks very much, Tom. I think we should move on to our next talk. Okay, so we will have another 2 talks, and then we'll have just a short comfort break. So just to let everyone know, that will be 5 minutes after the next 2 talks. Okay, so continuing on with the Wolbachia theme, we've got a talk from Steph Rainey about Wolbachia and lipids in Aedes this time. Thanks.
Thanks, Jeremy. And thanks, Tom, as well, for a great introduction, because it means that I can go straight into it without giving too much of an introduction. So yeah, I'm Steph Rainey, and I work in the Sinkins group in Glasgow. I'm going to talk to you a little bit today about how we are trying to determine the mechanism by which Wolbachia causes antiviral activity. So we're really lucky in the Sinkins group to have a large number of mosquito colonies with different Wolbachia strains. We also have a number of cell lines as well, which I can tell you, Tom, it doesn't matter if you put it in IEDs, they will still lift off the plate. So today I'm mainly going to concentrate on this section of Wolbachia strains here. And this is because we actually understand very little about how Wolbachia causes antiviral activity. But what we do know is, and what we thought for a very long time, was that density was really important. So the more Wolbachia you tended to have more antiviral activity. However, a couple of years ago, Tom in the lab managed to get a line in Mites aegypti, which only contained W. alba, which comes from Albopictus. And although it reaches a very high density, it's not able to block viruses very well. So we've used this to kind of try and tweeze apart what's going on. So a couple of years ago now, we showed that in cell lines that contained the strain WML pop, there was an accumulation of esterified or trapped cholesterol, which is what you can see over here. And this corresponded to a drop in the amount of free cholesterol within the cells. And this was really interesting because a lot of arboviruses require cholesterol or lipids in order to replicate. So we went on to look and see if manipulating the cholesterol within the cells, we could rescue the antiviral activity. And this is indeed what we saw. So we used a cyclodextrin called 2-HPCD, which you can use to increase or decrease the turnover of cholesterol within cells. And we were able to see at 3 different concentrations in the cells that contain Wolbachia, we could rescue dengue replication. So this was really interesting. We also have several proteomics data sets from midguts. So we published in 2017 that there was lots of perturbations in different pathways, but we showed quite clearly that there was perturbations within lipid metabolism. And this seemed to correspond to an increase in metabolism of lipids in the presence of WML compared to midguts that didn't contain Wolbachia. So we've since gone on to look at different Wolbachia strains, and what we've seen that's really interesting is that the strain WAU, which is probably the best blocker of arboviruses, actually doesn't show similar perturbations to WML. So what you can actually see here is that there is either no significant perturbations seen in the light colours, or it's in the opposite direction to WML. And W. alba, which sits, that has the high density but no blocking, sits kind of intermediate between the two. So this was really interesting to us. So we've gone on to look at this more in a cell physiological basis. So hopefully you can see this. So over on the your left is cells. So these are albopictus cells. And what you can see is that under a steady state, up the top where we don't have Wolbachia, there is, we've got top floor, which is a cholesterol derivative in green. And what you can see is that when there's no Wolbachia in a steady state cell, there isn't very much punctate staining. However, when you add 2-HPCD, you increase the turnover and you get more. However, in WML, we have the opposite, where we have a huge accumulation of cholesterol within lipid droplets. And when we treat this with 2-HPCD, this reduces, and this corresponds, you'll see in the next slide, to, again, an increase in viral replication. We can recapitulate this in midguts. So it might be difficult for you to see, but here you just need to concentrate on the very punctate red staining, which you can see here in WML decreases after 2-HPCD treatment. It's quite difficult because it's merged a bit there. So we looked at this in WAU cells and midguts, and again, the perturbations were different from WML. So down the bottom, you can see the cell lines where there's kind of similar levels to negative cells, and when you add 2-HPCD, it increases, and we saw the same in midguts. So as I said, we can rescue, in WML pop cells, we can rescue dengue replication by introducing 2-HPCD. So this is the general blocking that you normally see, and you can see AU tends to block a bit better than MEL. And indeed, in this case, this is Zika virus, and you can see that under all concentrations apart from 5 millimolar, we can rescue the replication of Zika, which is exactly the same as what we see for WML pop with dengue. However, if we do this with WAU, it doesn't matter how much we put on, it doesn't matter what cyclodextrin, we're not able to rescue replication at all. So again, it's suggesting that the mechanism by which AU is blocking is different from WML. So to try and figure out what was going on, we went back to our proteomics data set, and here what we decided to do was to do a sort of overall analysis. So instead of looking at single proteins, we looked at KEGG pathway analysis, and we compared our previous data set WML to our AU and our ALB. And the theory behind this was that anything that was consistent between all three strains probably wasn't antiviral because ALB isn't as antiviral. So what you can see here is this is WAU in the middle, and you can see that it causes many more perturbations than the other two lines, and that any that are similar between the three tend to be KEGG pathways that are important for general growth of Wolbachia. So we went on to look at this further, and we took out KEGG pathways that were significant only in AU, so not in ALB, to try and figure out what was going on. And what became apparent really quickly was that RNA binding protein pathways were significantly overrepresented in our KEGG pathway analysis. So this is what you can see here, and it involved lots of different things from RNA polymerase to degradation to ribosomal biogenesis. So we then worked a little bit with Alfredo Castello's lab in the CVR. So he works on RNA binding proteins in the context of viral infections, and he's able to basically carry out a RIC protocol, which is where you capture RNA, and once it's been cross-linked to proteins, then you pull it down, and you can do mass spec to see what proteins are definitely RNA binding. And this was really good for us because, as it is with all mosquito work, you'll know that although we know some things about proteins, we don't know everything. So what we were able to do with this was we were able to significantly increase the number of known functional RNA binding proteins. And what this showed us was it recapitulated what we'd seen in the previous analysis, that WAU has significant perturbations in RNA binding proteins that we don't see in ALB and MEL, and that this is very clearly skewed towards being upregulated. So why is this important? Well, as I said previously, lipid metabolism is really important for arbovirus replication, but so is RNA biogenesis and RNA processing. So I've just taken 3 genes out here, which we're currently looking at. So these are a dead box helicase and an ATP-dependent RNA helicase, and A Bravo, which is an uncharacterized protein. And these are important because they've been shown to be antiviral by other groups quite significantly. So you can see here that if you knock them down in cells, there's an increase in, I think this is chikungunya and dengue. And then Alan Cole's group in the CVR is currently studying A Bravo, which is the bottom one. And you can see that it is antiviral against Zika and chikungunya. And we're currently in the process of looking at these both in vivo and in vitro. And so far it's looking like they might have some effect on WAU's ability and also WML's ability to block viruses. So to finish off, so lipid metabolism is really important for antiviral activity of some strains of Wolbachia, but not all. And it's strain and virus specific, which hopefully you'll hear about more in Daniela's talk that's coming up. It also seems like RNA binding proteins are important, and it could be that these are important in both strains, but to different levels. And the main reason that we do this work is because if we have a better understanding of how Wolbachia strains block, and if we can determine that different strains of Wolbachia block in different ways, then it gives us a safety net if any time viruses were to overcome Wolbachia protection. So with that, I'd just like to thank everybody in the group and Alfredo's group for the RIC data, and I'm happy to take any questions.
Great, thanks so much, Steph. Any questions for Steph? Okay, I can start off. So in terms of the levels of Wolbachia, I guess these you referred to like mid-gut levels, or is that the whole mosquito?
So for ALB and for AU, the tissue tropism and density is very similar.
Okay.
So it's not that the algae has got less in the midgut or less in the salivary gland. Between AU and male, AU is a bit higher, and Maddie's going to correct me when she's nodding. AU is a bit higher in the salivary glands compared to male and a bit lower in the midgut. But with regards to the algae, it's not that it's just in the wrong tissue. It's in similar densities within the tissues that are important.
Okay, and but the blocking effect is happening in those midgut cells, basically.
It can, yeah. So we have data that it happens in the midguts and also in the salivary glands as well.
Okay.
Yeah.
Okay, great. Any other questions? Yes.
So AU in Drosophila is non-CI inducing, right?
Yes.
How does it perform in AEs?
It's non-CI inducing.
So that phenotype is. So what's the prevalence rate per generation? So it's maternally transmitted, and you get up to nearly 100%.
Yeah, we do. Yeah, we have to check the colonies regularly, and some backgrounds tend to lose it a bit quicker. But yeah, we tend to check it on most occasions. I mean, the one thing about AU that's interesting in Drosophila is that you can find it quite prevalent in wild Drosophila strains. But there's obviously a reason, right? Because it's not causing CI. So one of the hypotheses would be that it gives some kind of fitness phenotype, which could be blocking.
Protecting against viruses that kill Drosophila. Yeah, that's really interesting. Thank you.
All right. Any other questions? Okay, good. We can keep going then. Try and get on time. Thank you very much, Steph. Okay, great. So we've got another talk from the Sinkins group, Daniela Laferti, who's also going to tell us more about Wolbachia in Aedes mosquitoes and arboviruses. So over to you, Daniela. Thank you.
Hi, yes, I'm a postdoc in Steve Sinkins group. My name is Daniela Laferti, and yeah, as Steph has already covered a lot of the differences between the Wolbachia strains, I'm only going to just briefly mention that in these studies I've been using WML, WLB, and WAU. So, as most of you already know, I'm sure Wolbachia is really good at blocking positive sense RNA viruses such as alpha viruses, including chikungunya and SFV, but also flaviviruses such as Zika and dengue. And by positive sense RNA viruses, we mean viruses that have a genome that can basically act as directly as an mRNA, so it can be directly translated into a protein. But when it comes to negative sense RNA viruses, and there's not been a lot of Wolbachia studies made in looking into the effect that Wolbachia has on these types of viruses, and the studies that have been done have been a bit conflicting and also mainly focused on ISVs. But there's loads of medically important negative sense RNA viruses, including bunyavirus, Ngari, Batai, Rift Valley fever virus. They've caused several outbreaks over the years, one including bunyavirus here in Kenya in the 90s. They can also both affect humans and ruminants, so they're huge agriculture importance as well. And they're also massively underreported and often misdiagnosed because a lot of the symptoms they cause actually overlap with those of other mosquito-borne diseases. And when we say negative sense RNA viruses, we mean a virus that has a genome that needs to first be transcribed into an mRNA before it can be translated into a protein. And their genome tends to be segmented, so the replication differs a lot from positive sense RNA viruses. So I wanted to look into whether Wolbachia is able to block these negative sense RNA viruses. So first, what I did was I used our Wolbachia cell lines, so AA23 cells, and I infected them with bunyavirus, and I looked at 24 hours and 72 hours post-infection. And so I took supernatant and looked at infectious virus particles via plaque assays and also via RNA in the cells via qPCR. And as you can see, at 72 hours post-infection, there was a significant reduction in the amount of bunyavirus within these cells. However, I found it a bit weird that the drop is not actually as much as we tend to see with the alpha viruses or flaviviruses for that matter. So there is less than a log drop, but it was still blocking. However, when I looked at the Wolbachia density of these cells, I actually was surprised to see that Wolbachia had actually significantly dropped at both 24 hours and 72 hours post-infection with Bunya virus, which suggests that Bunya virus is actually affecting the Wolbachia density within these cells. And so I wanted to look into this further using our in vivo Aedes aegypti model. So I infected Aedes aegypti mosquitoes by feeding them an infectious blood meal containing 1 × 10⁷ of Bunya virus in blood. And then I looked at 9 days and 12 days post-infection. So I dissected out salivary glands to look at how much virus was present via plaque assays. And then also I took the carcass to look at viral RNA via qPCR. And as you can see, on overall, there was no blocking observed in any of the time points. Weirdly enough, there seemed to be a slight enhancing effect by WML at 9 days, and not at all at 12 days. And I repeated this experiment since, and I could no longer see this enhancing effect. So I think it's just randomly that this group of mosquitoes all ended up getting infected during this infection. But on overall, it's clear to see that Wolbachia can't block Bunya viruses in Aedes mosquitoes. And when I looked at the density of these mosquitoes, I saw the same thing as I saw in the cells. So there was a significant block in Wolbachia density in these mosquitoes, which suggests that Bunya virus actually reduces Wolbachia density within the mosquitoes of Aedes aegypti. So I wanted to look at another negative sense RNA virus. I used Rift Valley fever virus, and I did an infection in vitro, so using the same cells as before. I looked at 48 hours post-infection, and I saw no effect on Rift Valley fever virus infection at all. So I want to repeat this experiment in mosquitoes as well to see if Rift Valley fever virus also has a negative impact on Wolbachia density. And currently, I am in the process of doing 2 major experiments where I'm looking at the Wolbachia density in subsequent generations following a Bunya virus infection. And also I'm trying to look into the impact that this Bunya virus reduction of Wolbachia has on potential co-infections of positive sense RNA viruses. So for example, if you had a mosquito that had been infected with Bunya virus and then you see a reduction of Wolbachia, can it still block positive sense RNA viruses? And this is important because if we ever have another outbreak with Bunya virus or another negative sense RNA virus, we need to know if this can impact the density of Wolbachia that we see in our field populations. So to conclude the data I have so far, Wolbachia does not block infection with Bunya viruses. Instead, Bunya virus significantly reduces Wolbachia density within the mosquitoes, and this could have an important impact on the density in mosquito populations. And thank you all for listening.
Excellent job of keeping to time as well. Any questions for Daniela? Yes.
Great talk. Rift Valley fever is mainly transmitted by Culex mosquitoes, which are fully infected with Wolbachia. Would you think it has any effect?
It could be. It could be worth looking into if there's a difference between the mosquitoes.
Any other questions? Okay, great. Thank you very much, Daniela. Oh, question. Did I miss one?
Do you think like if you have an insect specific from Bunya family that can affect Wolbachia outside here in the field?
You mean an insect-specific virus?
Yeah, like
so there are a few other studies that have looked at the effect of Wolbachia on those insect-specific viruses, and depending on the ISV, sometimes Wolbachia blocks them, and sometimes it doesn't. So I think they act probably a little bit different, as it's not an acute infection.
What about like an insect specific from Bunya family?
Yeah, that's what they looked at.
Great. Thank you very much, Daniela. Okay, so we're going to have a really quick 5-minute break. So that'll be 3:10. I want to see everyone back in here. Okay, just an announcement. There's been a little bit of confusion, but if anyone wants to get coffee or snacks, we're going to go and do that now. So we're going to slightly extend this short break by an extra 5 minutes. All right, can everyone hurry up and take their seats? And we'd really like to get started. Okay, I think we will try to make a start. So if those that are still coming in could just quietly sit down. Next up, we've got a very exciting talk about bringing Wolbachia technology to Africa. So looking at Wolbachia in African strains of Aedes aegypti from Mavi Mancini. All right.
Good afternoon, everyone. I'll keep the conversation, the discussion on Wolbachia going, and especially about its application for vector control and dengue control in different endemic areas of the world. In 2016, we, in collaboration with the IMR of Kuala Lumpur in Malaysia and the University of Melbourne, we have started open field trials of Wolbachia deployed strategy. By releasing WLB carrying Aedes aegypti females. WLB is a Wolbachia strain that is naturally found in Aedes albopictus. And we got really interesting results, and the impact was very promising. After 3 years of constant monitoring of the populations that we have released, we observed that Wolbachia frequency was above 80% basically in all the release sites. So the population really established very well in all the release sites. But also the impact was at epidemiological level too. So we saw a decrease when we compared pre-intervention and post-intervention incidence of dengue over multiple years. We saw that there was an association of the presence of WLB with a reduction of dengue incidence, and we're talking about between 40 and 85% throughout all different sites. We are now in an expansion phase, we are now scaling up the program. The sites became 30 sites, and now the program is also in collaboration with the Ministry of Health. And also the impact on the dengue incidence is becoming like more substantial, and we got a reduction of dengue incidence up to 85% in all the release sites. So these are just the latest data; they are unpublished data. So the idea is now to investigate whether these interventions could be applicable also in other endemic areas of the world, such as the African continent. But of course, before considering any potential field translation, we have to consider a thorough and comprehensive characterization of the line that we're going to release, and also of the interaction between the symbiont, between the strain of the symbiont, even variants of the same strain, and the host background. And this is what we did. So thanks to the collaboration with the Diabate group at the IRSS in Burkina Faso, we managed to import in Glasgow a field-collected colony of wild-type mosquitoes. We took the male and we crossed them with WLB-carrying female from a lab-adapted strain where we have generated the first transinfection. We did this for several generations, and after 5 back crosses, we were quite confident to have a stable introgressed line. So we started to characterize the line. The first thing that we looked at was the intracellular density of Wolbachia in whole bodies and in different tissues of female mosquitoes. We compared the WLB infection in the Burkina background and in the lab-adapted strain. And as you can see from the plots, we didn't see any difference in the Wolbachia, any significant difference in the Wolbachia density in the whole females or in the ovaries, in the midguts, and in the salivary glands. We also wanted to assess the response of Wolbachia to high rearing temperature, high tropical rearing temperature, because we know that some Wolbachia strain might be perturbed by high larval rearing temperature. So we exposed the larvae to a 24-hour cycle of temperature recorded from the field, from Burkina Faso, and then we assessed the density of Wolbachia in the females that emerged from the larvae, and also here we didn't see any significant difference. And of course, we were interested in looking at the traits which are important for the applicative side. So we wanted to see whether the LB strain would be theoretically able to invade the population through the reproductive manipulation of Wolbachia that is called cytoplasmic incompatibility, and WLB. And we confirmed that WLB in the Burkina background is also able to induce CI. In fact, when we cross males carrying WLB with uninfected females, we saw that the progeny was unviable. And then we looked at vector competence of this new introgress line. We challenged mosquito with dengue 2 virus, and we look at the virus in the salivary glands, and we had in both the Burkina background and in the lab adapted background a substantial block of the amount of virus in the salivary gland, so which is a proxy for determining the ability to be transmitted. We also looked at other host fitness traits affecting population dynamics. We look at the survival, at the fecundity of females, so the number of eggs per female, and also the fertility. So we look at the number of the hatching rate of eggs after different time of quiescence. And also here we didn't see major difference between the WLB carrying mosquito and the wild type. For WLB in Burkina, you start to see a decrease. It's quite variable, but you start to see a decrease in the hatching rate after 50 days of quiescence. We also wanted to go a little bit deeper on the phenotype related to quiescence of eggs, and we look at the fecundity of females that emerge from quiesced eggs. We also introduce another group in this analysis, which is the Malaysia. So this is a recently field collected strain from Malaysia, from the field. And so you can see the number of eggs per female after different time of quiescence, or after different time points. If you compare wild type with WLB, you can see that there is a decrease in the fecundity of females in the Burkina background, in the lab strain background, after an extreme time point of quiescence, so after 6, 9, and 11 weeks, which is something that probably in the field is quite difficult to see. This doesn't happen with the Malaysia background, so the fertility of females is very stable no matter the quiescence of the eggs. The decrease in the number of eggs was also correlated to some morphological abnormalities that we saw on the ovaries. So we are now currently investigating these traits because, of course, it's important for the application of in the field. However, it is not a phenotype of WLB per se, but it's really due to the interaction between the symbiont strain and the background. Just a brief note that also our program is tackling, has the aim to tackle Aedes albopictus, which is a sympatric strain in Malaysia, and potentially is responsible of the residual dengue transmission that you have seen in the dengue incidence data. So we've just generated 2 new strains in the Aedes albopictus background with WMEL and WMEL pop from Drosophila melanogaster, and the idea is now to translate also the use of this line in the field very soon. So the takeaways and the conclusions of the talk are that WLB in the Burkina background displayed stable Wolbachia infection. It is able to strongly block dengue 2, and theoretically is also able to invade populations by inducing complete CI. We did have seen some impact at fitness level, especially in the quiescence, after quiescence of the eggs, but it's really related, we think that it's really related to the interaction with the host background. That's why it's really important for us to explore the divergences between host and symbiont combinations in order to develop the most effective dengue control intervention. And I want to thank the group in Glasgow and our collaborators, and thank you all for the attention.
Great, thank you very much, Mavi. Questions for Mavi? Yes.
I've got two actually. The first one is, you know, when you did, nice talk by the way, but when you did the introgression, have you actually looked to see if the level of genomic introgression matched the expectation? And whether there are any islands that are...
Yeah, we are in the process of characterizing it, if there are like inversions, which are like chromosomally, like what we see in Anopheles, for example, in Anopheles coluzi, that there is the inverse chromosomal infection. We are in the process of characterizing the level of introgression. Yeah, no, we haven't done it yet.
Okay. And then can I have another one? It's quite incredible, this thing of the quiescence. So are you saying that they quiesce for a long time, and then even though you feed them sufficiently, their fertility is reduced? Is that hatch rate, or is that clutch size as well?
No, it's the hatch rate. It's the fecundity of females that emerge from quiesced eggs.
Yeah. But it's in the hatch rate of those females.
Yes.
Not in the clutch size. There's no difference in clutch size of eggs laid from those females.
No. It's in the ability to hatch, to survive.
Any other questions? Yes.
Well, mine's sort of a follow-up question to Tony's first one on integration. I think they struggled when they released WMel in Rio de Janeiro because of insecticide resistance differences between the integress line and the wild type population. So is that something you're gonna maybe look at as well?
Yeah, we will. And of course, before considering any releases, the lab colony that has been characterized, it will be anyway refreshed with a wild type colony. This is what we did in Malaysia. We continuously refresh the background, and the main reason is because of the insecticide susceptibility. That apparently is lost after 6 generations from what we have seen in the Malaysia.
Right. Okay. Any other burning questions? Okay, yes, one back there. We'll have one more, and then we'll move on to the next speaker. Yeah, one. Okay, good.
Thank you. I heard that for Wolbachia, you need to screen the mosquitoes at some point to know whether it's like 100% contaminated. Are you checking whether it's like 100% contaminated? Infected, sorry.
Yeah. So, for example, for WLB, the maternal transmission is 100%. So there is very high fidelity in the vertical transmission. So we do check our colonies, but it's just for quality control. So periodically we want to check the prevalence, but usually we have very, very, very high infection rate.
So do you mean that even if it's not 100%, when it comes to upscaling and releasing in the field, you don't really need to screen?
I mean, you can do it as a procedure for quality control, but I mean the maternal transmission for this strain, for example, is very stable, and it's always 100% from what we know, at least for now.
Okay. Okay, good. I think we'll end there. Thank you very much, Mavi. Just a quick general announcement. I know it's all very exciting stuff and you want to take it home with you, but can we limit taking pictures of slides with data? Some of that's unpublished, and it's better that you talk to the people before sort of taking a picture of the data. So limiting that. Thanks. Right. Next, we've got James McNamara, again from Sinkins Group, will tell us about cytoplasmic incompatibility.
Cameron.
Oh, sorry, sorry. Okay, sorry, Cameron. Yes, Cameron McNamara from the Sinkins Group, tell us about cytoplasmic incompatibility in Wolbachia. Over to you, Cameron, thanks.
Okay, so you'll have heard a lot of the background on Wolbachia from my colleagues and Tom Walker. So the main takeaway is that it's a maternally inherited intracellular bacteria that can manipulate host reproduction in order to spread. In mosquitoes, this manipulation mechanism is called cytoplasmic incompatibility, or CI, and it occurs when Wolbachia-induced modification of the paternal chromatin disrupts early rounds of mitosis in embryos, leading to lethality, unless rescued by a compatible Wolbachia infection. So CI is mediated by two co-diverging genes, which are referred to as the CI factors, or CIFs, and these CIFs can be seen to interact in a cognate-specific manner of binding, and there are many cognate pairs in Wolbachia, and they can be divided into 5 phylogenetic groups, and type 4 CIFB genes are expected to encode 2 functional nuclease domains, and this nuclease activity is expected to be important for CI induction. So CIFB has largely been associated with the induction phenotype and CIFA with the rescue. However, depending on the CIF homologue or insect species studied, CIFA can sometimes be seen necessary for CI induction, which has led to diverging ideas on the mechanism of CI. So in the toxin antidote model, it's hypothesized that CIFA inhibits CIFB toxicity during spermatogenesis, and then upon transfer to the egg, CIFB can modify the paternal chromatin unless the antidote CIFA is supplied by a Wolbachia infection. The alternative host modification model suggests that CIFA assists CIFB in modifying the paternal chromatin during spermatogenesis, and that neither CIF peptide is transferred to the egg. Rescue by CIFA expression is expected to be dependent on either the reversal of the modifications on the paternal chromatin or by making compensatory modifications on the maternal. So to inform this debate, I wanted to test CIF gene expression in an alternative insect species, and because CI mediation is important for the Wolbachia release programs in Aedes aegypti, I wanted to test in this insect species. So I've tested a few CIF genes from different strains and different types, but in this presentation I'm going to focus on the type 4 CIF genes from WLB, based on this strain's relevance to the Wolbachia release programs. So firstly, I expressed CIFB in the testes and found that it induced complete sterility when transgenic males were crossed with either uninfected or infected females. And dissection of the mated spermathecae from the females and dissection of the testes and seminal vesicles revealed that these tissues were devoid of mature sperm. So because type 4 CIFB peptides are expected to act as nucleases, I expected that DNA damage might be occurring, which would disrupt spermatogenesis, leading to this spermless phenotype. So I performed tunnel assays, which label DNA breaks and nicks in green, and you can see that there's abundance of DNA breaks in the transgenic testes. So next I thought that CIFA might inhibit this toxicity, so I expressed CIFA in the testes and crossed them into the CIFB lines to see whether this would rescue sperm production. And in certain genetic combinations, this was seen to be the case and was dependent on whether CIFA had either higher or comparable levels of expression than CIFB. Furthermore, although they produced sperm, the transgenic males were still incompatible with uninfected females. However, infected females were able to significantly increase the viability rate of embryos, which suggested that this sterility was based on CI. So looking at the testes of these double heterozygotes, I could see that although sperm production had resumed, there was still a large abundance of DNA breaks, which might have suggested that CIFA was localizing CIFB to specific target sites. However, as you can see in the WLB infected testes, there's no DNA damage occurring, which suggests that the modifications associated with CI do not occur during spermatogenesis, which conforms to a toxin antidote model of CI. But this also suggests that perhaps the balance of the CIF peptides in our transgenic system has not quite been met. So to resolve this, I wanted to express both CIF peptides at near equimolar quantities under the same promoter using a T2A sequence. And I found that these lines were able to induce rescuable CI. However, the rescue capacity of infected females was rather low. So next I wanted to fiddle with the dosage in the males. So I either increased the CIF-A dosage in the males by crossing in the strongest beta-2 CIF-A line into the T2A lines and found that this increased the rescue capacity of infected females. And next I lowered the dosage, the overall dosage of both CIF-A and B by swapping the beta-2 promoter for the weaker TOPI promoter. And this was also found to increase rescue capacity. So based on the toxin antidote model, rescue is associated with CIF-A binding to CIF-B in the embryos, and therefore CI strength can be associated with the level of deposition of CIF-B via the sperm. So lowering the overall dosage with the TOPI promoter makes sense as to why rescue is increased, but it begs the question why increasing the level of paternal CIF-A dosage also increases the rescue capacity. So I hypothesized that either CIF-A is relocalizing CIF-B from the paternal chromatin before transfer, or that excess CIF-A bound to CIF-B could inhibit the CIF-B toxicity after fertilization. Next, I wanted to test whether maternal CIF-A expression could rescue CI induced by the transgenic males. So I linked CIF-A expression with the maternal X2 promoter and found that it was able to significantly rescue CI induced by transgenic males. So this represents the first recapitulation of both induction and rescue phenotypes in transgenic Aedes aegypti. And although the results seem to mirror that of the ALB infected females, the rescue capacity of these X2 females is significantly lower, which might suggest that the level of maternal deposition is not quite enough to inhibit the CIF-B transferred via the sperm. But I expect that expressing CIF-B with the TOPI promoter and CIF-A with the beta-2 promoter would increase the capacity of rescue from the X2 CIF-A females. So based on ours and others' work, we can assume that the CIF peptides function in a toxin antidote model. And as TA components are often incorporated into gene drive designs, we can envision how a CIF-based gene drive system could function in Aedes aegypti. There's several benefits to a CIF-based gene drive, namely that Wolbachia-mediated CI is seen to function in a wide range of insect hosts. So these CIF-based gene drives could function in a large amount of vector species, which could prove beneficial in the vector species that have proven harder to transinfect with Wolbachia. And furthermore, there's an exhaustive range of CIF pairs, and these CIF pairs can have different enzymatic capabilities. So therefore, we have a wide array of TA components, which would be beneficial in cases of host resistance to a certain CIF drive that's released. So yeah, these are cool ideas that I'm having, so I'd like to continue on this. And I'd like to thank my group for everything.
Great, thank you very much, Cameron. Excellent. Any questions? Yes.
Just the thing you mentioned at the end there about resistance. In terms of natural examples of resistance, are they well documented if they're there? And would you expect them to be acting through the CIF genes as their targets?
There doesn't seem to be much resistance to CI. It usually evolved, I would say, the only kind of resistance is mutations in the Wolbachia that stop CI from happening, but I don't think that's a host effect. I think that's just an evolution thing. But yeah, like I said, there's different types and different dynamics. So if one was to somehow be, if the host was able to resist it, we could probably swap it for something else.
Okay, any other questions? Maybe just
Very quickly, how long did it take you to generate these transgenic lines?
Not very long. I'd like to say it was hard, but I think I generated like 86, 89 lines in 3 years I was working on it. Yeah. So it's really easy with the piggyback in Egypti. I mean, the hard thing is characterizing and looking after these lines, but it's pretty.
Great. Thank you very much, Cameron. I think we'll move on to the next talk. But yeah, thanks again. Great. So moving on to Waka Orsain, who's going to tell us about Tsetse fly microbiome. So looking forward to your talk. Over to you.
Thank you. I am Kalmes Bwaka from Crete, Cameroon, and I will be talking on assessing the tsetse fly microbiome and the potential association of some bacteria taxa in trypanosome establishment. So, trypanosomiasis is a parasite diseases, neglected tropical diseases, caused by a protozoa called trypanosome, which is transmitted by a blood-sucking insect, tsetse fly, to human and animals. In human, the disease is known as sleeping sickness, and in animal, the disease is known as Nagana. There were large-scale epidemic of sleeping sickness in the 20th century that was brought under control through the large-scale program of screening, diagnostic, and treatment. This program led to significant reduction of the disease over the time, with less than 1,000 cases reported today. That is why WHO considered the elimination of the diseases by 2030. However, no vaccine is currently available. Some drug, or the majority of drug used, are toxic, and the potential animal reservoir ensure the circulation of the parasite at relative low level, making difficult the elimination of the diseases. That is why we are thinking that reducing the parasite transmission ability of the fly can impair disease transmission and help for disease elimination. As we have been listening since this morning, different or much more studies are focusing on the bacterial community of the insect and their potential use for symbiont-based vector control. Therefore, understanding the role of the tsetse bacteria symbiotic fauna in disease transmission could improve knowledge in initiative to develop these new vector control strategies. That is why we aim to assess the microbiome composition of the whole tsetse fly and their potential association with flies' vector competence. Specifically, we identify the different trypanosomes circulating, as well as mature infection, and identify the different bacterial community present in tsetse fly and their association with trypanosome establishment. This work was conducted in Campo, situated in southern Cameroon at the border with Equatorial Guinea, and there are around 5,000 inhabitants, with main activity being farming, fishing, and hunting, exposing them to tsetse fly bite. And as you can see, cases of sleeping sickness are still reported today. So tsetse fly were collected using pyramidal trap, followed by morphological identification, and then the species Glossina palpalis fly were selected for the studies and separated in leg, head, and body. Head and body was used in PCR after DNA extraction for the identification of trypanosome circulating and mature trypanosome infection. For the identification of bacterial community, DNA was extracted from different groups of tsetse fly and then sent for sequencing of the V4 variable region of the 16S RNA gene of bacteria, followed by metagenomic analysis. About 18% of the fly was infected by at least one trypanosome species, with the presence of Trypanosoma brucei gambiense, the species responsible for sleeping sickness. And from this infected fly, 21% was mature infection, suggesting that these fly are really competent for the disease transmission. 31 bacterial genera were identified with Glesworthia being the most abundant, which is the obligate symbiont of tsetse fly. But these bacteria seem to be replaced by Burkholderia or Serratia in some Glossina pallicera or some Glossina tachinoides flies. Also, the abundance of Wigglerworthia in this study is actually low compared to other studies where the abundance was ranging from 19 to 99%. So this could be explained by the fact that we use the whole body to do the microbiome rather than using the midgut only, suggesting that other tissues of the fly are key location for the bacterial development. There were a statistically difference in bacterial community according to tsetse fly species. That means the different bacterial composition, there is a different bacterial composition between genetically distant tsetse fly population. And this should take into account if you have to do some kind of study for a vector control based in symbiont. Also, blood meal significantly increased the bacterial diversity, as we can see in the map, in the graph. And more importantly, Wigglerworthia, when there is, during the process of blood meal, Wigglerworthia tend to decrease, giving opportunity to other bacteria to be increased. And this could have certain role for tsetse fitness. There were no significant difference in the bacterial diversity when comparing tsetse fly harboring or not a trypanosome. However, and more importantly, in tsetse fly infected with trypanosome, there is a significant difference when comparing the one which is having a mature infection and the one which is having only the infection in the midgut. This suggests that the maturation of the trypanosome infection in tsetse fly depend on the composition of the bacterial fauna, or the trypanosome modify the tsetse fly bacterium in order to establish themselves. Looking deeply in the difference observed, differential abundant testing show many bacteria associated with the infection stage of the fly, as you can see in the table. So further study are really needed to understand the role played by this bacteria, and also maybe the mechanism, and if they could be target for the symbiont-based vector control in the tsetse fly. In the conclusion, 31 bacteria genera were identified with the primary symbiont Wigglerworthia being the most abundant. Significant differences were observed in the bacterial diversity of life with and without mature trypanosome infection, suggesting a change in diversity and composition during infection and maturation of the trypanosome. And we also see a strong association observed with certain bacteria with trypanosome status. And these bacteria need to further study to really understand the mechanism underlying this pattern. So I would like to thank this organization supporting the study, thank all my supervisors, and thanks for your kind attention.
All right, thank you very much. Any questions? Question.
Hi there. You said that there was a strong association between the species that you said and trypanosome presence, but was that association positive or negative?
Yes, thank you. There are some bacteria associated negatively and some associated positively. So both need to further investigate it to really see the mechanism and if you can target the bacteria for some vector control use.
Yeah, just here. So the effect you see of change of diversity after trypanosome infections, do you think it's what could be causing this? Is it an indirect effect from the host?
I think the change we observe in diversity when the trypanosome is maturation, as we say, we have two hypotheses. Whatever there is a change in microbiome diversity in tsetse fly for the trypanosome to be established, so the trypanosome actually modulated the microbiome to establish themselves. or the trypanosome need a certain bacteria composition in such a fly to get inside the gut of the fly. So these are the two hypotheses, and we really need to further investigate to really know what is the real.
One more question, yeah.
Thank you for the presentation. I just wanted to ask, you said the microbial profiles were different, and you attributed this to maybe
that could be really important. So it is important to look the whole bacteria composition and then target the one and go and study where it is located. And if it is important, we can continue the study.
Okay, great. Thank you very much, Hussein. Excellent. Right, so shifting now to sandflies, we've got Damaris Matoke Muhia, who's going to tell us about endosymbionts and gut bacteria in sandflies. So over to you, Damaris.
Yeah, as you've heard, I'm Damaris Matoke Muhia. I'm going to speak on sandflies. I think since yesterday, we've not had anything on sandflies apart from mosquitoes, and I think my previous speakers who managed to diverse from mosquitoes. So here I'm going to report the work we did on exploring whether sandflies contain endosymbionts and the role of those as far as transmission of leishmaniasis is concerned. Here are the collaborators we've been working with. So to start with, sandflies are the main vectors known to transmit leishmaniasis, which is one of the neglected tropical diseases. Especially in Kenya, we have visceral leishmaniasis, cutaneous leishmaniasis, which occurs mostly in arid and semi-arid areas in the country. The disease mostly affects people from poor resourced communities who have challenges to access health, treatment, and also management as far as getting commodities is concerned, even testing kits. So also in those areas, especially in Kenya, issues of conflict are an issue whereby we have nomads or pastoralists who tend to have conflicts to do with their livestock. So you find there is a lot of movement around, therefore having fixed facilities becomes a challenge, or even reaching them out as far as vector control activity is concerned is also a challenge. The disease is also aggravated courtesy of lack of targeted vector control that targets sandflies, and more so there is no vaccine towards that. Treatment mostly needs inpatient, whereby someone has to be in hospital for around minimum 17 days, which becomes also a challenge. Here I'm showing a map of Kenya on how this disease is distributed. Visceral leishmaniasis is quite rampant in most of the counties in Kenya, but cutaneous is also now becoming an issue of concern. So having challenges, considering even there's lack of mapping where the vectors are, there's quite a lot of habitats that favor occurrence of this disease. Recently, the Ministry of Health, the NTD, reported emergence of those cases in areas they don't even expect. And also even the treatment, people are not responding to treatment regimens that are there. Therefore, there is need for us to start thinking what are other solutions apart from testing and treating, but also vector control initiatives. So we need to be innovative on how to control the disease. So our question in this study was, can endosymbionts be exploited in controlling leishmaniasis in Kenya? Now we don't have much data globally that documents a presence of microbiomes in sandflies. So we went out to explore this. So this study was part of the bigger study that was being done by various institutions in Kenya, and we were able to collect sandflies from 3 counties, but also further, which are in the Rift Valley region that have both visceral leishmaniasis and cutaneous. And in this, we also screened sandflies that we have in our colonies in the insectaries at Kemri and Isipe. So we, after trapping of the sandflies, we were able to do morphological identification of these flies. But further, we also were able to do PCR so that we are able to do subspecies identification. For the parasite, that's the Leishmania parasite, we dissected the sandfly, the midguts, to check presence of the amastigotes. But also we did PCR targeting the ITS region to be able to confirm what parasite it is. But further, we did sequencing to confirm the subspecies. We didn't do sequencing for all the samples, but just a subset of samples just to confirm what we had identified. Further, for us to be able to screen, to understand the endosymbionts, we screened the sandflies for just 5 major endosymbionts. That's Wolbachia, Rickettsia, Spiroplasma, Cardinium, and also Microsporidia. We used PCR for this and sequencing. As far as sandfly, for my results, as far as sandfly species population is concerned, we were able to trap around 1,700 sandflies. Of these, they belong to around 60 species, with the main one, which are the Phlobo sandflies, being 6 of them. That's the Phlebotomi gugis bagai, and then we have Martini, Orientalis, Duboski, and Aculators. These, the phlebos are the main vectors that transmit leishmaniasis. We also screen Sargentomias to just see how different it is from phlebos that transmit leishmaniasis. So as far as presence of endosymbionts and any other gut bacteria is concerned from the field collected, most of the sandflies trapped from the field had Wolbachia. But of concern is Gugis bagai, which is known to transmit cutaneous leishmaniasis. We found Wolbachia, but also there was Rickettsia, and also Phlebotomi martini, which is known to transmit visceral leishmaniasis. It carries Leishmania donovani. Also, there was Rickettsia on that, and Phlebotomas severs, which also had Rickettsia. So as far as looking at the colony sandflies is concerned, we didn't get most of the Wolbachia and Rickettsia, but we were able to pick Serratia marcescens, which is known to be very toxic in aphids, and it's been used for controlling aphids infestation. Therefore, we didn't explore much to understand it for the colony raised. But further, we were concerned that the presence of this, what does it mean? However, the sample size was very low because it was just 8 sandflies out of 17 that we screened. So we realized that most of these sandflies had various microbes. And when we went into further looking at just the phlebos and leishmania interactions, we realized that phlebos add mostly of the main ones, that's Wolbachia and Rickettsia, and then this needed further investigation. So as determining whether this was significant, any sandfly that was infected with leishmania, that's mostly Leishmania donovani or Leishmania tropica, we found that it was also infected with Rickettsia. So therefore, which meant that there is a role that this Rickettsia is playing as far as the Leishmania parasite development in the sandfly is concerned. So this is just to document that presence of Rickettsia in infected sandflies with Leishmania parasite. So we also looked at what are the subspecies of this rickettsia. We found that most of them had Rickettsia africae and also other species, which we are now exploring further to sequence to understand whether these are significant or they have a significant role as far as development of the parasite is concerned. So in conclusion, we are saying that sandflies tend to have different endosymbionts, including Rickettsia africae, which is known to be causing febrile disease rickettsiosis. But also conclusion is that it's likely that any sandfly that had leishmania will be infected with rickettsia. Can this be a potential vector control tool? So I'd like to thank the support we received, the funding from ANTIVEC and also the various collaborators, Greg Hurst from Liverpool, Cambridge team, Ministry of Health, and PAMCA. Thank you.
Thank you very much, Damaris. Any questions for Damaris?
Yes, thank you for your presentation. You mentioned that Rickettsia africae was detected in sandflies. So I'm just wondering that does this translate to mean that the Rickettsia is transmissible? Is transmissible by the vector?
I think those are amongst things that we are exploring right now, and that's why I said that is ongoing. It's likely to mean that, or it has a role to enable the parasite. For us, we're looking at the leishmania, so to enable the parasite to develop in the sandfly.
Okay. The other thing I'm just curious about is how you manage to have a colony of sandflies. I understand that they breed on decaying matter, right? So how do you do that?
Yeah, we have had the colony for a while now, and we just imitated the conditions in the field, in the insectaries that we have. We have various species of the sandflies in Cambridge.
Excellent. Okay, one more quick question, Thomas.
Do you think by colonizing them in the lab could be the reason why they might have lost rickettsia?
Maybe, yeah, because that has been generations. It's been for a while, so it's likely that's why they lost. But we are currently collecting new and trying to screen to see whether those can be without or with. Yeah, check it.
Okay, great. Thank you very much, Damaris. Okay, so we've got 2 more talks today. We'll try and hopefully finish in time for the bus. They'll wait a little bit. Okay. Right, excellent. So next we've got Etienne Bilgo from Burkina, who's going to tell us about transgenic mosquito-killing fungi. So over to you, Etienne.
Thank you so much, Jeremy. Good afternoon, everyone. So my talk is going to be on the transmission of transgenic fungi during copulation. I want first of all to say thank you very much to ANTIVEC for giving me this opportunity to present this recent data that we just generated in our lab. So unlike bacteria and viruses, fungi are infected mosquitoes through contact. So they are working like chemical insecticides. So unfortunately, my slide is on the roof. But what I want to emphasize that you can see here the fungi the spores on the fungus, on the cuticle of the insect, going through the different layers of the cuticle before. Okay, maybe, let's see. Okay. Yeah, everything is on the roof. So, so it takes around 3 to 4 days for the fungus to reach the emolength. And when the fungus is gonna reach the emolength, it's gonna take around a week before killing the mosquitoes. So one of the landmark paper in this area using fungi to control mosquitoes was published in 2005 by Scott and collaborators, and they have shown in Tanzania by a simple design, by impregnating the cloth with the fungus and put them in the roof of houses in rural area in Tanzania, they can reduce the number of infectious bites from 250 by 52 per year. You can see here in this picture in the left, some mosquitoes touching the impregnated cloth, and they can be infected, and then they're gonna die from the infection. But since this paper, we didn't make a lot of progress in terms of using this fungi for operational control of vector in the fields. So there are a lot of difficulties around that, but one of the most important issues when it's come to use this fungi to control mosquitoes is the weakness of killing mosquitoes. They're taking around 10 days to 2 weeks to kill mosquitoes. That means that mosquito, even they are infected by the fungus, they have enough time to go to mate before yeah. Okay. Okay. That means even mosquitoes are infected by the fungus, they have enough time to go to bite people, transmit the disease before dying from the infection. And also they have enough time to go to mate, to lay eggs, and contribute to the next generation before dying from the fungal infection. So our idea here, as this fungus is really specific to mosquitoes, is to use the fungus to speed up the virulence of the fungus by making him expressing some toxins like the Androtonis australis toxin from a spider. And this is I won't take a lot of time to explain this process, but it's the idea here is to use the fungus like a vector in order to reach the emolength and release different kind of toxins that we want to use. So the rationale, the basis behind this bite transgenic system using fungi, that we can use different sources of genes. We can use genes from the host insect itself. Here it's the mosquitoes. We can use genes from the fungus itself in order to improve the virulence of the fungus. We can use also genes from the predators like this gene here that I'm gonna talk later about that. And we can also artificially synthesize some genes in order to improve the virulence of the fungus. Here you can, for the rest of my talk, I'm gonna talk a lot about this gene, this toxin called hybrid toxin, and this toxin is acting at the two-channel level, at the calcium level, and at the potassium level. When you compare that to the paratrinoid insecticide, they used to act at the sodium channel. That means that we are using a toxin which is acting at two channels at the same time. And this gene is really well characterized, well used in the US, for example. You can use that without any approval. So we have generated a couple of in collaboration with St. Leger Lab at the US, and we have shown in one of our papers that we can even combine different genes in the fungus. So we can design the system in order to allow the fungus to express different type of genes at the same moment. For example, we made this combination, hybrid toxin and AAIT, and we have shown that in one of our papers that only one spores of mosquitoes is able to kill mosquitoes because the spore need only to reach the emollient and release the toxin. And we have also shown that this system, the transgenic fungus, is really working perfectly with the insecticide. I'm gonna go back a little bit here. For example, if we are using a transgenic fungus with the insecticide, like for example, the paratrinoid, we're gonna act at the 3 channel at the same time, calcium, potassium, and sodium. So it's gonna be really hard for the mosquitoes to develop resistance against 3 channels at the same time. So after that, we have shown in Burkina Faso in one of our seminal work that Diabaté was talking about yesterday, that this is working very well at the mosquito sphere. And we have shown by releasing males and females through different generations that only we need the transgenic fungus can collapse the mosquito population by only 2 generations. That means around 43 days. So now I'm coming back to my population recent work. So as the fungus is working like chemical insecticide, they are infecting mosquito through contact. You can imagine that if a male is infected by the female, if a male is infected by the fungus, during the mating, he can transmit some spores to the females. And as we have shown that only one spore of the transgenic fungus is able to kill mosquitoes, we don't need a lot of spores to kill mosquitoes. This was the idea. And we developed this project with this rationale to use them in combination also with SIT, classical SIT or GM SIT approaches. So what we have done, we did the infection like for chemical insecticide with WHO cylinder tubes, impregnated cloves, put them in the cylinders and expose mosquitoes to the fungus. And you can see here in the green dot the legs of mosquitoes with some spores. And then we did in the lab some artificial mating. In our jargon, we used to say force mating. I don't like that. I want the term artificial mating. So we cut the head of the male, the infected male, and make them inseminate a female. And during that, we counted the number of spores that can be transferred during this system. And we have shown that on average, the wild type or the transgenic fungus can transfer around 9 spores to the female. And then we follow the mortality of the females. We found around 90. The survival here in the green, this is for the hybrid one. The survival was around 90. The survival of the transgenic one was less than 20%. That means 90% of mortality. So then we went to the mosquito sphere. We released 250 infected males against 500. We released 500 fungal infected males against 250 females in the mosquito sphere, and we let them swarm. During the swarming, we observe the behavior of the mosquitoes, the number of swarm per compartment, the size of the swarm, the number of couples in the swarms. And then we can see here in this map, we use 4 compartments. This experiment, we ran that on 10 replicates, and we have here the compartment, and we have the number of couples per swarms that you can see here. And we found that we found around 24%, 24% of the females are mating with the males. That means around 60 one couple per swarm. And then we follow the mortalities. We recapture the mosquitoes and we follow the mortalities in the lab. What is really interesting, I want you to focus on the figure here. We have a mortality which is around a survival which is around less than 75%, but that means that the hybrid kill around 26.7 mosquitoes. This is really interesting because it's matching really well with the 24% of couples that we have seen. That means that with the hybrid, all of the couples that made, they really die during the copulation. This is a key message that I wanted to show during this talk. So in terms of implication, we have shown that fungi can really work very well with the mating-based system like SIT, and also I didn't show this result, but it looks like we need these windows of 24 hours after infection with the fungus to see this system to work. And also only 9 spores able to run this system. And at the end, we think that also we can use fungi to study mating behavior system for mosquitoes. Thank you so much, and sorry for my slides on the top. Thank you.
Thank you very much, Etienne. I think even if you change the name, you're still sexually abusing those mosquitoes. Okay, any questions for Etienne?
Thank you for your talk. It was very interesting. I would like to know how you manage to attract the male to the infection point.
Yes, what we used to do, you know, in our team, I'm talking with a lot of modesty. We are, our first expertise is mating. So we know how to manipulate mosquitoes and let them mate. So in mosquito sphere, we used to release the ratio one female for 2 male. So that means in the experiment we release 500 males against 250 females, and we release them at the right moment during the sunset. And you can see in the mosquito sphere because we make the environment mimicking the outside environment, and you will see the mosquito mating, and you will see the couples going out of the swarms. Yeah. No. We did that with the WHO tubes. Just put them, yeah, before releasing them. Yeah.
Thanks, Etienne. This was really cool. So in the application side of these things, if you had these sort of cloths to put on the roofs of the homes, how often would you have to change them? Like, how long do these fungi live?
Yeah. This is a very important question. So with the transgenic fungus, we have shown that it can last for 3 months. Yeah, but because for the transgenic one, we just need only one spore is able to kill because they are living stuff. We need to make sure that they're still alive. So, but from our experience, like it's like 3 months. Yeah, but for the wild type, it's taking around 1.5 months, something like that. Yeah.
Okay, great. Yeah.
Talking to some of the collaborators in China, they seem to have, a specific technology where they can help, to come up with a sheet. This is going to be impregnated, and then it can last for 2 years.
Great. I think we'll move on to the next talk, but thank you very much, Etienne. Excellent. Okay, great. So last talk of the day, but last but not least at all. So we've got a talk from Pauline Nana. He's going to tell us about metarhizium and its use against ticks. Over to you.
Thank you, Jeremy. My name is Pauline Nana. I'm an entomologist working with the University of Ebolova in the south of Cameroon. But this part of work was done at TCP in Nairobi. So the title of the presentation is The Virulence of the Wild and Genetically Transformed Strain of Metarhizium anisoplia against cattle ticks. It's almost the same as the previous presentation, but this time on ticks. So the livestock rearing is an important source of income for many small and large-scale farmers around the world. The development of the livestock faces a certain number of challenges, and one of the most important is the infestation by ticks. Ticks are the most important ectoparasite of livestock in tropical and subtropical area. Losses due to ticks infestation are estimated to about $19 billion annually, and over 800 million cattle are constantly exposed worldwide. Ticks cause losses through direct effect of blood sucking, and heavy infestation, as you can see on board, cause dermatitis, anorexia, and many other ailments. But by the same process of attachment and feeding, ticks also transmit various pathogens and toxins, leading to diseases such as theileriosis, babesiosis, and aplasmosis that has been reported to kill more than 1 million cattle annually in Tanzania alone. So how do the farmers deal with this threat? Farmers mostly rely on synthetic chemicals and repellent. Although the acaricides have been shown to reduce tick population and their incidences, the main drawback is that they are considered as environmental pollutant. There is the risk for milk contamination, and more importantly, ticks have developed resistance to these various classes of compounds. This is the status of tick resistance in the whole world, and you can see that in East Africa, it's very serious. So this has prompted the search for new alternatives, such as biological control. And among candidates of biological control, we have the entomopathogenic fungi that has been reported to kill the host through the cuticle and do not require injection to be active. And this component is very important when dealing with blood-suckers arthropods such as flies, mosquitoes, or ticks. But the slow acting of EPF or entomopathogenic fungi have prompted the search, or have prompted some researcher to engineer the genome of the fungus by introducing the scorpion toxin, neurotoxin gene to increase their virulence. So the objective of